Welcome to the BioStock studio and the first ever spring edition of our flagship event, the BioStock Life Science Summit. Over the next 2 days, we, together with our partners Nasdaq and Porter Clarkson, will bring you more than 20 exciting presentations from Nordic life science companies, as well as panel discussions with industry experts. But without much further ado, let's get started. First out is Gasporox and CEO Märta Levander-Schie. Welcome.
Thank you so much for that introduction. And yes, my name is Märta Levander-Schie and I'm the CEO of the company Gasporox. And what we do is quality inspection of containers of pharmaceuticals, food and beverages. So what we do and bring to the industry is a fully automated inline inspection of these containers. We do pharmaceutical, food and beverage containers, and we have the products of sensors that we provide directly for inline integration to our machine builders, and instrument that we sell through a distributor network. Our products are based on a patented laser technology that makes it fully non-destructive and means that you can have an automated inline control. We are listed on First North Nasdaq and we are located here in Lund. We are a team of 15 people. Our focus is of course sales and get our products and technology into the market. Within the company there is also a broad technology competence, and we have, for example, built the company GPX Medical that was divided out of the company. We also have a project on porosity and material analysis of pharmaceuticals. But let's talk about why do we do what we do. We all know that we are a growing population of 10 billion people that will reach by 2050, and all of us use a lot of pharmaceutical, food and beverages. And it's seen that it creates a lot of food waste, and almost 30% is discarded even before reaching the store. We also know that 67% of Sweden's population collect prescription medicine. And 135 million injections per day are made from glass containers. And these are numbers before the pandemic, so you can imagine how these have risen the last period. And all of this creates a lot of waste within the quality inspection. And one thing that has been shown to be very efficient is the use of modified atmosphere. That gives a sustainable production and a safe production, which is key. Looking into the importance of modified atmosphere packaging, it has been seen as one of the most effective ways to reduce food waste and improve packaging, for example, by using modified atmosphere packaging. But what is modified atmosphere packaging? It's about the gas inside the packages. We talk about oxygen, we talk about carbon dioxide, we talk about nitrogen. These are standard gases that are present in ambient air, but having the correct gas mixture of them really gives a longer shelf life and a safe product. And what is important then is that you have the correct gas mixture and that you have a tight package. They're talking about package integrity and leak testing. And this is exactly what Gasporox technology can provide a quality measure of. So if you think about, for example, fresh pasta, but not maybe all of you are aware that this is packaged in a modified atmosphere. But an open package has a 3-day shelf life, while an intact package has a 1-month long shelf life. This is the same within injectables, for example, IV bag, where it's very oxygen sensitive products and often packaged in a primary package and a secondary package with an oxygen absorber around it. And this shows the importance of having it really dependent on the gas mixture inside. And it's about sterility, but it's also about the correct gas mixture. And that's exactly what Gasporox technology can provide information about. And we have then modules where we can measure both in pharmaceutical, food and in beverages. And it's all everything from vaccines within pharmaceuticals to within food, it's meat and ready meals, and beverages, it's wine and champagne. These are all big segments. You see food being the largest, but looking at the product price of each of them, pharmaceutical of course has one of the highest. But what is interesting is that these are all segments that we can reach. They are controlled by different regulations and directives like USP and FDA. One especially interesting part is the parenterals. that has a value of $500 billion US, which is a large portion of the total pharmaceutical market. And here it's really critical that our injectables that gets tested, and this is one of our key applications. What is interesting with Gasporox product within these segments is that we use the same product to reach them, but we cannot reach these ones all at the same time. So we have decided to do them year by year here. So now having a pharmaceutical focus, then moving over to food and then beverage. But the product is the same. A little bit about product inspection today. Typically in a factory there is a lot of automated inspection. We have check weighers, X-ray, vision system with cameras, etc. But the gas and leak integrity is typically very manual. And you have to have these control points, both in food and pharmaceutical. It's quite highly regulated. And we have reached so far that within pharmaceutical, it's actually even mentioned our technology with laser headspace analysis, especially then on lyophilized vials for container closure integrity testing, which is a really shows that our technology has a really benefit and need in the industry. And that's also why we have now put a lot of focus on the pharmaceutical industry. What about the test methods today? These are typically destructive in nature, extractive with needles, operators using instruments that extract the gas. Manual testing, it gives a lot of waste. It's inefficient and unsafe production, while the tested product does not go out to the consumers or patients. Looking at the Gasporox method, you get a fully automated inspection of both the gas mixture and integrity. And the ones who doesn't pass, they are rejected directly online. It's automated, it's no unnecessary waste, and it's cost efficient, and it's a safe production. This gives 100% control. We have it in 3 product lines: the gas spec that measures gas inside the package, we have the leak spec that measures the gas who leaks out of the package, and we have the instrument series where we also have put some emphasis the last year to also offer for lab and at-line testing. The business model is divided in 2 segments: the sensors that we sell to machine builders for integration that then sells to producing factories, while the instruments are sold directly and through a distributor network to a producing factory or lab. Through this we get both an expansion in segment and in geographical expansion. Giving an example, like the sensors or products are produced here in Lund, and we provide them to an integrator somewhere maybe in Europe. And by them providing it on a global scale, we can then with one customer reach on a globally, which is ideal as a small actor. Adding then each different segments on pharmaceutical, food and beverage and different partners within each segment, you realize that this is a very scalable business model that we as a small local actor still can maintain good customer support and still reach globally. Similarly, we have in our distributor network where we also work on a global area scale, where we produce in Lund and we have the instrument then are provided. Focus areas now are Asia, North America and Europe. There we have a pharmaceutical network and also a food network. These are not always the same. We have a scalable business model that enables us as a small player to reach globally and to many different markets. Looking at our strategy, we talked about the business model with long-term agreements and distributor network. I talked about the segment expansion where we now keep a focus on pharmaceutical, then moving over to food and then beverages. We're doing the geographical expansion. We have a target of 10 to 20% of newly installed lines annually. This is now reviewed for each segment where we also now know we can reach instruments and also reach already existing lines. But our focus is continued growth and always to keep technical innovation. And as I mentioned there, we have built the company GPX Medical from history. That was divided out of Gasporox in 2020. We also have a project on material analysis that we are seeing on how to incorporate in our business, where we last year we became a finalist in the CPHI Pharma Award, a very prestigious award. So this shows that we have really interesting potential also for other aspects of our technology that we are always taking care of. Looking at the finance, looking at the 2020 year, we grew by 21%. We had a turnover of 12.5 million, and we had a negative -4 million EBIT. We closed on a cash of about 24 million. We have a positive sales development, but with the pandemic impact. Our expansion strategy affects operating profit, but we are really segment by segment moving forward here. Looking at the first quarter numbers, we had a turnover of 3.3, 7% up from last year. They continued focus on market establishment, the pharmaceutical focus. We also got a Vinnova grant. We still experience a continued impact from COVID-19, that is traveling restrictions, and also especially on food segment we see that we have a troubles, more difficult to do some integration projects. This share you can see from the last year here, a big increase when we did a dividend of GPX Medical, but we have a stable ground. We have 8.6 million shares, 1,200 shareholders. We have a market cap of 120 million Swedish crowns. We have a very competent board, the chairman Per Nyström, also the owners represented through Christophorius and Fora Capital, and also Jan-Erik Smith from Gobia Enterprise. Looking on the future now, 21 to 25, we have been in a phase of a pre-market where we have sensed where are our solution best in the market, and now we really feel that it's the pharmaceutical focus where we feel the market pull, and we need to go on this one. So now we're doing the market establishment, and we do this by focusing the strengthening of sales team. We have an experienced and senior team. They have several years of experience in technical sales to the pharma industry. We have them located in Central Europe, and we also do investment on these systems and modules. And we are further recruitments ongoing and planned during 21 and 22. The plan forward is the segment expansion, pharma, food, beverage, as I mentioned. We have the geographical expansion, Europe, Asia, North America. We have the complete modules for faster distribution, standalone instruments and modules. And this is also part of the pandemic where we cannot travel and do the integration project as we typically have done. This means the instrument series that we have expanded. The latest one being our GPX 1500 Film Pharma that was launched just a couple of weeks ago. This is a unique product where you for the first time can measure non-destructively in IV bags. We have had it tested at pilot customers and got very positive feedback. It was just launched in connection to PDA Parental Packaging Conference, and we are now excited to see how the market responds to this one. This is what we call then an instrument, which is used for at-line testing or lab testing. And you can then easily measure the oxygen within such a pharmaceutical bag. Looking further then in 2021, we have secured the long-term growth, of course, in focus, strengthening the sales team, as I told you, more integration customers, sales of instrument, a geographic expansion where we have Asia in focus. We do the instrument and modules for easy integration and fast distribution. We have an expansion in pharmaceutical market, and that's, for example, the launch I just presented to you, and always to review our technology potential. And we have the focus that all packaging with modified atmosphere shall be inspected with Gasporox products and solutions. And why should you then invest in Gasporox? We work in several large growing markets. We have a unique technology. It's a patented product. We have a leading position with this solution. We have a clear and strategic plan, and we work for a sustainable and secure production. And we have a technology with big potential. So with this, I thank you for listening and open for question.
Thank you so much. Like you said here, the solution can be applied in a wide range of industries, but the focus will be pharmaceuticals. Just to sum up, why did you decide to focus on the pharmaceutical industry?
We have had a couple of years now where we have done what we call pre-market, test a couple of different solutions in these pharmaceutical food and beverage segments. But what is very clear to us is that pharmaceutical is one step ahead when it comes to like quality control and inspection. And there we have got to the level where our technology is well known. If you visit a pharmaceutical conference and you talk about the HSA method and CCIT testing, they all know what you're talking about. We get a lot of requests on this. So to follow up on that request, we have then decided to have that focus. In parallel, it's also been that we have been blocked by an exclusivity that is going out to this September now. So this is also like we have not been so well known in the pharmaceutical industry, and now we say we need to build our own brand in this to take the full potential. And that's why we're adding the instruments, we're adding IV bags, we're looking into ampules, etc. So there's a lot of things also to be done within the pharmaceutical that we understand is maybe easier to reach in the short term and compared to the food and beverage. I would also say that the pandemic has had an impact where we see that the food and beverage are a little bit more business as usual, that they're up and running, while pharmaceutical is really pushing forward and taking like many leaps forward now. And I mean, you see many new actors also popping up, and also you need this inspection then part of it.
Yeah, so it would be a good opportunity to get into that market.
Yeah, and I would say that we are in that market, so it's more about taking
the next
step and taking it to new segment. Like now we reach certain, but we maybe don't reach Asia as much. So, for example.
And you did mention here that you have patents for the products and so on. What does the patent situation look like, and what's the strategy in that area?
We have a clear patent strategy in the company where we have, first of all, we have quite many broad patents. We have a patent family of 7 or 8 patent family, and then we have a strategic way to do that. First we go to Sweden, PCT, and then US, Europe, typically Japan. And then we have sometimes more on the technology, maybe then it could be on specific product, it can be a specific package format. So we always have quite open eyes on how can we patent these things. And we've been quite successful in getting our patents approved.
And then finally, you did tell us a bit about the plan for this year, but what is the most important thing for you to achieve in the business during 2021?
I think the pharmaceutical instruments is a key for us this in 2021, but also to become the brand within the pharmaceutical industry, because we have not been well known there, and we are taking more action on that. We had planned to exhibit at the CPHI last year, but it was cancelled. Now it's postponed to the fall, so we're very excited to be present there. But also we participate in digital events, we have posters, we try to, with the instruments it's also easier to get the more direct contact with end users. So it's a lot about building Gasporox as the pharmaceutical company that we actually are, that maybe not everyone is aware of.
But we look forward to following you then.
Yes.
And thank you so much for coming.
Yeah, thank you so much.
Our next presentation comes from Cyxone and CEO Tara Heitner. Welcome, Tara, and please go ahead.
Thank you, Celia. Well, it's good to be back at BioStock. And for those of you who are unfamiliar with Cyxone, we are a company that is really focused on patient quality of life. We're dealing with disorders of the immune system, chronic diseases, as well as acute diseases like COVID-19. Now, for patients with these types of diseases, they can look very healthy on the outside while their immune system is ravaging them from the inside, and it can be a very painful and lonely journey. So at Cyxone, we're focused on developing first-in-class disease-modifying therapies that deliver efficacy but without compromising the patient's quality of life. We have 3 programs in our pipeline, with Rebexmod being our lead candidate, which is currently in a clinical trial, phase 2 clinical trial for COVID-19. And that's very exciting. It's progressing well, with already 10 sites in Eastern Europe recruiting patients. We're also getting prepared for a very exciting phase 2b trial in rheumatoid arthritis, and I'll say more about that later. Then finally, we have ramped up our activities in T20K, getting ready for IND-enabling studies that'll prepare us for a very good entry into the clinic for multiple sclerosis. So first I'll start talking about our program in rheumatoid arthritis. Now, why do we need new drugs for rheumatoid arthritis? For patients who suffer from this disease, it's a progressive, chronic, lifelong disease, and current standard of care is currently unacceptable. Patients are treated typically as a first-line treatment with methotrexate. And while this is a drug that works, it only works in about 70% of patients. 33% of patients typically don't respond to the drug, and 25% of patients don't tolerate it well, so it causes severe gastrointestinal problems. Often the disease progresses, and then you have to escalate to very complex treatment regimes. And as the disease progresses, it requires changes of doctors, changes of treatments, as a constant race to keep the disease in remission. What our vision is for Rebexmod is that we can have an early treatment that can replace methotrexate or be on top of methotrexate to lower its dose, and be a simple treatment regime that patients can take for a long time before the need for treatment escalation, perhaps even preventing the need for treatment escalation. Now, why do we think we can do this? Rheumatoid arthritis is a disease where the damage to the joint is caused by a subset of immune cells called infiltrating inflammatory macrophages. These are normally friendly, but in rheumatoid arthritis patients, they become hostile, attacking the joint, destroying the tissue, the cartilage, and the bone. But Rebexmod is a drug which has a unique mode of action, specifically targeting the subset of immune cells, changing them from a hostile to a non-hostile phenotype, protecting the joint from damage, and stopping the progression of the disease. Now, very recently we had some news that we have a very exciting collaboration initiating with a leading rheumatologist called Professor Constantino Pitsalis. And this is also validating our approach and the mode of action of Rebexmod, because he has worked with many RA drugs and believes that Rebexmod, with its unique mode of action, preventing damage to the joint, can have a place in a rather crowded RA field. So we still have plans to start our trial at the end of the year, and this will be in a subset of patients that we will believe have a response to Rebexmod. So we're narrowing the patient population, and we would also take biopsies of patients, so we'll get some insight into what's happening in the joint and how the drug is working. The trial will read out around 2023. So again, this is a very active field, but it's a very big market, many patients. And with our drug, if we are replacing methotrexate or having an alternative, as well as delaying the need for biologicals, we believe we could command a healthy slice of this market It would be a new market, approximately at peak $5 billion US. Now, if I talk about Rebexamod in COVID-19, we're all quite aware that this pandemic has lasted much longer than we expected. And even with the advent of vaccines, we need to have therapies to treat patients. Indeed, even very recently on May 6th, there's a new EU strategy was revealed where they support the development of vaccines, but also put a large effort into the development of therapeutics. Why are we developing Rebexamod for COVID-19? Well, it's very similar mode of action. These viruses, when they infiltrate the lung, could be COVID-19, but this is also applicable to other viral infections. They cause a macrophage to infiltrate the lung, and in COVID-19 patients, these macrophages become overactivated, releasing cytokines into the lung. And the cytokine storm can lead to sepsis, and this can lead to organ failure and eventually death. Rebexamod, again, because of its unique mode of action, can prevent this overactivation of the macrophage, preventing the release of cytokines and preventing the need for patients to be hospitalized. This is our vision. If the trial is successful, we would consider a fast route to market, an accelerated route to market for Rebexamod in COVID-19, and it is something we're pursuing, but we're also looking at alternative paths forward. The trial, as I mentioned, is in progress, and we expect the readout to be at the end of Q3 as planned. And we're very excited because we've been very diligent in opening up many more sites than originally planned in order to recruit patients in a timely fashion. And this is going along quite nicely. So now, finally, our exciting program in T20K for multiple sclerosis. So as you know, multiple sclerosis is a debilitating disease that attacks the nervous system. The immune system attacks the myelin sheath of axons in these patients. And it can occur suddenly in very young people at a time of their life when they're going to start new careers or families. So it progresses all through life and can end up with people needing to be in a wheelchair. T cells, or immune cells, are the culprit, and these cause demyelination of the axon. It affects about millions of patients, 2.5 million patients globally, and many of these cases are called remitting relapsing multiple sclerosis. The symptoms are varied, and people can have cognitive problems, tingling in the extremities, suddenly falling down, or sudden blindness. Right now, the treatments that are available can slow the disease sometimes, but it doesn't work across all patient populations, and there are serious side effects. So patients are demanding better solutions. T20K is a drug which has been shown in animal models of multiple sclerosis to halt the disease progression when applied at an early stage. It actually acts on the immune cells by reducing the cytokines IL-2 and the expression of IL-2 receptor on immune cells. And this is a target or a cytokine that is very well known to be responsible for the progression of multiple sclerosis. So we are currently moving forward with our preclinical IND-enabling studies, and we have initiated studies on biomarkers, and this will have the effect of making our clinical trials more predictable and simpler. We're also looking at alternative administration forms, and this will give us more options going forward and lower the risk of the clinical trial, give us more chances on goal. Again, multiple sclerosis is a large market, and with T20K, it's a novel mode of action. It can command its own market with the opportunity for over $5 billion in sales at peak. So this is our pipeline revisited And we have as our business model to partner our programs at proof of concept around phase 2 stage of development with strategic partners who have the capability for later stage development as well as commercialization. And for Rebexamod, we are in the process of reaching out to potential partners. It's going very well. So we have many milestones to look forward to throughout the year. Right now we are in a warrant period, and we are excited to encourage warrant holders to exercise their warrants. We also have the trial, the RA trial preparation ongoing, as well as our collaboration with Professor Pitsalis. As I mentioned, many preclinical studies on and IND-enabling studies for T20K that'll position us for a very strong phase 1 clinical trial. We expect the readout of the COVID trial at the end of Q3. In the fall, we also have in Q4 the Rebexamod patents, which will be public, as well as our T20K patent going public. And we're very much active in getting the company prepared to uplist to the main market in 2022. And finally, in the long run, we see this company with an expanded pipeline with drugs close to market or on the market, and a much larger team with the bandwidth to really push our programs forward. So with that, I'd like to say thank you for your attention and happy to take any questions.
Well, that's good because I have a couple of them for you. Thank you for the presentation. You mentioned here the collaboration with Professor Pitsalis. What will that mean for Rebexamod going forward?
Well, first of all, as I mentioned, Professor Pitsalis is an expert world-class rheumatologist with his focus is on biopsy-driven and innovative clinical trials for RA. Believes that in order to develop innovative therapies, we need to do clinical trials more clever and smaller, not so big and expensive. And he thinks that that is the reason why there's not a lot of innovation happening in RA. So it's really great for us as a small company to work together with him. It also validates Rebexamod as a mode of action, that there is indeed a need for this in the RA market as he sees it, having been exposed to working with many RA drugs that are on the market. And finally, the biopsies that we will introduce into our trial design will give us, it will be like a little camera inside the joint that will tell us how our drug is working and validate our mode of action early on.
And moving on to your other candidate, you have also announced news regarding T20K.
Yes.
What are the main differences between the old plan and the new plan for the candidate?
Yeah, that's a good question. Well, we originally had a vision for T20K as an oral therapy, and we maintain that vision. We're still going forward with that. However, we're also looking into additional modes of administration, including a sub-Q, and that would be like a simple cartridge similar to what they use for insulin. And by having these two different approaches, it just gives us more opportunities for success. We know that both forms are showing efficacy in animal models, but we don't know in a patient which one will give the greatest effect on their disease, have the greatest safety and convenience. So we want to make sure we can deliver the best thing for the patients. We're also looking at biomarkers, and that's something new. And what the biomarkers will do is allow you to have an indication of whether your drug is working without having to run very long trials where you have to wait to see a sort of biological effect. Because long trials are expensive, and as a small company, you want to be efficient. And so we want to be able to tell if our drug is working in a shorter amount of time so that we can run a more powerful trial.
And let's finish on the financial side of things.
Yes.
You have the subscription period for the TO4, which is ending soon.
Yes.
What is your message to people who might still be considering to subscribe?
Yes, I think it's a good opportunity. I welcome all to exercise their warrants. The warrants that are not exercised will expire without value. So in order to not lose their value, there needs to be active subscription for the shares. and the time for doing this is short. So before May 28th, which is in 2 days. So you should contact your bank for instructions on how to do this, or look at the instructions on our website, cyxone.com.
Well, thank you, Tara, so much for coming.
Thank you for inviting me.
And our next presentation comes from Alligator Bioscience and newly appointed CEO Søren Bregenholt. Welcome, Søren, and the floor is yours.
Thank you, Cecilia. Yes. Hi, I'm Søren Bregenholt. I'm the incoming CEO of Alligator Bioscience. Alligator Bioscience is a public biotech company developing tumor-selective immune therapies designed to specifically activate the immune system in the tumor and not elsewhere in the body. Therefore, our second generation immune therapies combines maximum efficacy with minimum toxicity, thereby differentiating from the first generation immune therapies that are currently on the market. A look at our forward-looking statements. Let's start with an overview of Alligator Bioscience. Our two lead products, mitazalimab and ATOR-1017, henceforward 1017, addresses two key immune activation pathways that are both important for immune responses to human cancers. Both programs have enormous medical and commercial potential and comes with strong preclinical and clinical data packages that support their best-in-class potential in their respective indications. And Alligator Bioscience is underway to move both of these projects into clinical phase 2 trials later this year. Alligator has a strong innovation track record, having brought a total of 5 products all the way from discovery into clinical development, both on our proprietary technology platform and not the least on our key in-house drug discovery and development competences. As a testament to our innovation capacity, we recently developed a third generation immune therapeutic technology called NeoX Prime that allows a much more personalized tumor or immune therapy treatment, and I'll talk much more about NeoX Prime towards the end of this presentation. As I said, Alligator Bioscience is an immune oncology company. We are focused on developing drugs to address significant medical needs in human cancers. Immune oncology drugs like checkpoint inhibitors have really changed the cancer treatment paradigm and helped millions of patients over the last decade. Although these first generation molecules are very effective, still only 1 out of 5 patients respond in a satisfactory way. And Alligator Bioscience's goal is to increase the response rate by developing second generation drugs with very high efficacy and a low toxicity profile that allow us to engage in combination therapies with checkpoint inhibitors, with chemotherapy, and other standards of care. As already mentioned, Alligator Bioscience is exploring 2 candidate drugs within 2 key immune pathways that we are taking into clinical development, and those are mitazalimab targeting CD40 on so-called antigen-presenting cells, and 1017 addressing 4-1BB on T cells. Both of these programs, as I said, address clear unmet medical need and hold strong commercial potential. Let's first take a look at 1017. 1017, as I said, is an antibody against 4-1BB, a protein on T cells, and very important for activating T cell immune responses to cancer. The target 4-1BB has recently seen emerging clinical validation, which of course increases our belief in our molecules being effective in the clinical trials that we are currently undertaking. So 1017 is a tumor-selective antibody that activates T cells and thus have significant potential to work in synergy with checkpoint inhibitors like PD-1. And you can say that the mechanism of action is like PD-1 is believed to release the brakes on the immune system, while 4-1BB pushes the gas, really allowing for a strong combination cocktail there. 1017 is now in late phase 1 clinical trials, and we expect to present the first set of data from that trial later in the summer in connection with the ASCO conference. We believe that 1017 is the optimal 4-1BB monoclonal antibody. Here we combine it with two previous antibodies, two competitors. On your left you have urelumab from BMS, which is a very strong and efficacious antibody, but comes with a very strong and unfortunate toxicity profile that has been limiting the clinical dose to 8 milligrams. And on the right side, you have an antibody from Pfizer, which has been sort of overcompensated and has a relatively weak effect on T cells and is only sort of activated under perfect conditions and limiting its clinical efficacy. Whereas we believe with the unique properties of 1017, we have a very strong antibody with a strong efficacy profile and a very beneficial safety profile, which is supported by preclinical data. And the next slide, I will show you how these antibodies are designed to have this optimal efficacy safety profile. So the key design element here is that 1017 is requiring cross-linking for being activated. That means that both ends of the antibodies needs to bind simultaneously, so at 4-1BB on the T cell and at the FC receptor at the antigen-presenting cell, the macrophage. And as you can see on the left side of the slide, in the bloodstream, in the circulation, there is a high level of unspecific immunoglobulins that bind to the FC receptor on the macrophages, thus not allowing cross-linking of 1017, although it's bound to 4-1BB on T cells. However, in the tumor, as you can see on the right side of the figure, the level of immunoglobulin is low, thus allowing 1017 to bind to 4-1BB on the T cells, while at the same time binding to the Fc gamma receptors on the macrophages, thereby allowing cross-linking of 4-1BB on the surface of T cells, leading to the activation of and efficacy and effector function of these cells. This is further illustrated on this slide, where we look at the ratio of tumoricidal CD8 T cells to regulatory T cells. And what we can see on the left side of the figure is that we have a significant increase in this ratio, which is predictive of a beneficial tumor response in tumors that are treated with 1017, where on the right side of the slide, you'll see no such change in ratio or activation in the periphery, sort of underpinning the specific cross-linking dependence of the molecule. So taking a look at our molecule 1017 in relation to competitors, there are a couple of antibodies out there in phase 1 and phase 2 clinical trials. What we can see is that 1017 is differentiated based on a high efficacy by combining a very optimal binding to the so-called domain 2 of 4-1BB on the T cells, supported by both clinical and preclinical, in vivo and in vitro data, and then the tumor selective activity by the need for cross-linking. And we can see that we have a dose range for the molecule that is very beneficial. Now let's take a look at the clinical trials currently ongoing with 1017. So as I said, 1017 is in late phase 1 clinical development. We have cleared the 100 milligram dose and is currently assessing the 200 milligram dose. We've so far seen only few drug-related adverse events. We hope to be able to read out at least interim data in June and in relation to ASCO. And then hopefully we can start a phase 2 study, potentially in gastric cancer, later in the year, with a readout for combination data with PD-1 antibodies or PD-L1 antibodies in the second half of 2022. Now let's change gear to mitazalimab. Mitazalimab is again an approach to try to increase the response rate in oncology patients. Mitazalimab is a CD40 agonistic antibody that has the potential to increase the efficacy of checkpoint inhibitors or immune we do mitazalimab again, okay? Thanks. Now let's change gears and talk about mitazalimab, our CD40 antagonistic antibody, which has the potential to increase the efficacy of checkpoint inhibitors and chemotherapy and other types of standard of care. So CD40 is expressed on antigen-presenting cells, such as macrophages and dendritic cells. Both are crucial for T-cell activation in the tumor. And mitazalimab improves the antigen-presenting capacity of these cells in the tumor, which in turn leads to T-cell activation, T-cell infiltration, and finally T-cell mediated killing of the tumor cells. Our preclinical data, as I said, support strong potential for mitazalimab to increase the efficacy of both PD-1 antibodies and chemotherapy. The phase 1 study has been completed with more than 100 patients assessed, giving promising signs of single-agent efficacy. So again, mitazalimab is designed for high efficacy and low toxicity to allow for combinations with checkpoint inhibitors, chemotherapy, as I just mentioned. And again, this is achieved through targeting the optimal binding epitope on CD40 in combination with the requirement for cross-linking of the molecule, as I just described for 1017. There are a number of competitors out there that also depends on Fc gamma cross-linking, but mitazalimab is the only one to be dosed at the top of the dose range of around 1 milligram per kilo, and hence we believe that the molecule is highly differentiated. And here on the next slide we can see a significant or an overview here of mitazalimab with its nearest competitors, again focusing in on the ability to dose very high of the molecule due to the high efficacy-safety ratio. And we really believe that this will differentiate mitazalimab in the clinic. So pancreatic cancer and CD40 agonism. Pancreatic cancer is the third leading cause of cancer-related death in the US, and thus being able to treat pancreatic cancer will of course constitute a significant benefit to the patients and also a significant commercial opportunities. And we believe that being able to establish proof of concept in the phase 2 trial will clear a path towards registration and market introduction for mitazalimab. CD40 in pancreatic cancer was recently further validated by data that will be presented at ASCO, showing that combining CD40 with chemotherapy increased the 1-year overall survival rate in combination with or in comparison to historical data. And evenly encouraging, these trials showed enriched T cells in the tumor, activation of macrophages, and also positive alteration in the tumor stromal. Optimize-1, our phase 2 trial, is designed to take full advantage of this, both on activation of macrophages and stromal alterations, and we believe that the superior efficacy safety profile of mitazalimab will allow us to dose higher, use a more efficacious chemotherapy, thereby boding well for a positive outcome of these trials. Just taking a brief look at Optimize-1, we will combine mitazalimab with the chemotherapy FOLFIRINOX In pancreatic cancer patients, initially we will assess the safety of 2 doses with FOLFIRINOX, and once we have established an efficacy signal, we will combine with PD-1. And as I just said, this trial is really optimized to take full advantage of the profile of mitazalimab. We expect to start the trial in Q2 this year, reading out the first handful of patients in Q4, and then with a full data readout in the second half of 2022. So as I promised initially, I will just round this presentation off with introducing you to Neo-X-Prime, our new technology and a new concept within immuno-oncology. It's a bispecific antibody concept, so it's allowing antibodies to bind to 2 different molecules. It's based on CD4 or CD40, and basically Neo-X-Prime allows us simultaneously to bring patient-specific antigens to the antigen-presenting cell, thereby activating T cells in a patient-specific way, and at the same time activating CD40, as we just discussed, thereby giving a much stronger immune response. And you can see that illustrated here in the lower left part of the slide. And this is actually very efficacious. On the right-hand side of the slide, you can see that when you combine a CD40 and an EpCAM antibody as 2 single entities in blue, you have a relatively weak response. But when you combine these in our Neo-X-Prime format, you can see you have a very strong tumor-directed immune response. And we really believe that this is a promising 3rd-generation immune therapy. To that end, we recently signed and announced a collaboration with US biotech company Macrogenics, where we have embarked on a joint collaboration to discover and develop one novel Neo-X-Prime molecule against 2 so far undisclosed targets. And we are of course looking very much forward to the outcome of this trial or this collaboration. But more to come in later presentations. So finally, just summing up, Alligator Bioscience is a very, very exciting stage with 2 programs moving into phase 2 clinical trial later this year. 1017 is in late phase 1, and we hope to present encouraging data at ASCO in the beginning of June, and hopefully take that molecule into phase 2 later this year. And mitazalimab, our CD40 agonist, we will start phase 2 studies in pancreatic cancer in Q2 this year, and hope to have the first interim readout already towards the end of 2021. So thank you for your attention.
And thank you for the presentation.
Oh, you're welcome, Cecilia.
Like I think both of us mentioned, you are a newly appointed CEO for Alligator.
That's correct. First workday, June 1st, yeah.
Wow. So what attracted you to Alligator?
So Alligator has as you know, a long history. It has its 20th birthday later this year. What really attracted me was a couple of things. I mean, first of all, immuno-oncology is very important. It's important for the patients. It's at the forefront of innovation, and it's also a significant commercial opportunity for those that are successful. But what really attracted me was the strong pipeline. We talked about two molecules here, mitazalimab and 1017. And then behind that, a strong early pipeline, both of technology, Neo-X-Prime, but also a few other molecules like 527s and others that we haven't discussed today. And then thirdly, it's just a very, very talented team that backs all this up, and I'm really looking forward to getting my feet on the table and sink my teeth into the work.
Yeah, and if we turn the question the other way, what do you think attracted Alligator to you? What do what do you bring to the company?
Yeah, but I think that is a very good question. I think I bring a mix of a lot of things. I've spent the last 20 years both in pharma and biotech, so in antibodies and small molecules, in operations, in strategy, in research. So I think I bring a sort of mix of that knowledge and understanding across the value chain. I've worked globally, so I think I bring an international mindset of how to lead and how to engage. And thirdly, I think I'm sort of a go-do kind of guy. I think we have a lot of things to do in Alligator. I'm sure we'll be successful, and together with the team, we will make it work.
And to round off, I'll ask you a really difficult question.
Yeah.
If we end by looking ahead a little bit, where do you see Alligator in a couple of years? Let's say 2 years.
Yeah, I think that's a very good question. I think Alligator has to have, and of course I don't have all the details yet as I haven't started. We need to focus on being able to develop these molecules all the way to the patients. I think that needs to be our aspiration. That's going to be hard. That's going to be bumpy. So I hope that we will be towards the end of these trials, maybe start planning into phase 3, potentially with a partner on one of the two molecules. And then I really hope that we've been able to advance the earlier part of the pipeline and really leverage the value that I think is in Neo-X Prime. And then having built the team a little bit stronger and bigger than it is today.
Well, it looks like you have an exciting journey ahead of you then.
I sure do.
Well, thank you so much for taking time.
Yeah, you're welcome. Thank you.
For our next presentation, we are joined by Respiratorius and CEO Johan Dott. Welcome, Johan, and the stage is yours.
Thank you very much. I will present Respiratorius and our leading assets to improve treatment of aggressive lymphoma as well as COPD. Respiratorius was founded in 1999 as a research spin-out from Lund University, where the initial aim was to develop new effective drugs for treatment of respiratory diseases such as COPD and severe asthma, which then was an area lacking new effective drugs. And 20 years later, or more than 20 years later, the fact is the same, that COPD lacks effective treatments. In 2012, Respiratorius acquired the rights to the asset VL-001, and since then we have progressed the project through initial clinical studies, phase 1 and 2A, as well as we have been granted orphan drug designation and have approved patents in the most important markets for the asset. And we are now preparing the asset for a phase 3 study. Within respiratory diseases, we have now the project RESP 9000, which is now being prepared for a phase 1 study. If we dig a little bit deeper into VL-001, which is for the treatment of diffuse large B-cell lymphoma, which is the most common type of non-Hodgkin's lymphoma. And annually about 60,000 patients are diagnosed with diffuse large B-cell lymphoma. And the overall survival, in terms of 5-year overall survival, is about 60 to 70%. 10 to 15% are primary refractory to the first-line therapy, which is an immunochemotherapy called R-CHOP. And 40% of those who actually respond to the first-line treatment relapses within 18 months. You can see also that the medication markets for non-Hodgkin's lymphoma is increasing annually, and diffuse large B-cell lymphoma accounts for 30% of the total non-Hodgkin's lymphoma market. VL-001 is a combination of valproate and prednisolone. And in order to find a synergizer to immunochemotherapy, a new cell line model was set up, and different synergizers was evaluated. HDAC inhibitors are known to have known as a synergizer to chemotherapy, as it makes the DNA more vulnerable to chemotherapy. Valproate is a broad HDAC inhibitor, and also has positive effect on gene expression as well as cell cycle arrest. Our phase 1 data also show that valproate increases the expression of CD20, which is the target for rituximab, which is R in the R-CHOP chemotherapy. Valproate has been used since the 60s to treat epilepsy, and then has a well-known safety profile which significantly reduces the development risk. And we, in the phase 1/2a study, we used the same drug as for treating epileptics, and that was a significant improvement in time instead of developing our own drug. I also highlight prednisolone, which potentiates the effect of valproate, and then in the treatment plan, prednisolone is administered simultaneously with valproate, and the treatment pre-treatment starts 2 days before entering into the chemotherapy. The results from our initial clinical studies was very strong, and the results are having a significantly improved survival and without any significant side effects. As a comparison, we used data from the Swedish Lymphoma Registry for patients treated between year 2000 and 2015. And we're showing that we have a more than 10% increase in overall survival after 2 years. Another statistical analysis of the same data set shows that we have a 80% reduction of risk of mortality compared to R-CHOP alone. And this data was shown to EMA, which confirmed that Respiratorius may move VL-001 directly to a phase 3 study. And also we need pharmacokinetic results, since we have been focused on developing a new formulation of VL-001, or a new formulation of valproate, which is the API in VL-001. We also performed based on results from the initial clinical studies, we also performed a health economic analysis showing that taking into account the 10% increase in overall survival from 70 to 80%, we have performed a willingness to pay analysis, indicating that the willingness to pay per treatment is about $80,000 US. And running the calculus with the annually 60,000 new cases, the total potential market value is estimated to $5 billion based on the willingness to pay. However, as I said, we have developed a new formulation dedicated for the new use of valproate. And the formulation is a combination of immediate and extended release in the same capsule, and we just recently performed a issue of shares to support and finance the pharmacokinetic study of the new formulation, which will be a phase 1 study in healthy volunteers, and the data is required for entry into a phase 3 study. So far, a CRO has been selected, who will then be responsible for submission of protocols and documentation and regulatory approvals. The new formulation is important in terms of supporting the attractive pricing and realize the global market value potential of the project. And this is something that we have had consultation with price experts, and the new and the PK study may support such a market value. We have also performed an analysis of competitors, and what we can see in respect to adjuvant treatment to R-CHOP, and we see that we have a overall survival and lack of significant side effects with VL-001, which really stands out in the competition. There have been some high deal activity within the diffuse large B-cell lymphoma space. However, most of these assets are related to immuno-oncology assets, where diffuse large B-cell lymphoma is only one of several oncology indications. What we can see as value-enhancing milestones during the next couple of years is continuing the strive to making the project ready for a phase 3 study. And that is the only sensible strategy, as we can see, as any deal or any agreement will include milestones from the continued clinical development and approval of the drug. And in order to move the asset through a phase 3 study, we need an application to start the phase 3 study, and then we need a confirmation from EMA or FDA on the study design, as well as we need to start the PK study, which we hope to do during the second half of this year. We have also high hopes on the patent approvals. We have a patent application which is not yet granted, and we can also foresee that we will file new patents on the formulation as it is more or less ready for production, or parts of the production has already been started. If we then jump to RESP-9000 project, it is concerning a novel and innovative first-in-class anti-inflammatory and bronchodilator for COPD and severe asthma. COPD is one of the most common and rapidly growing diseases, and is characterized by inflamed and constricted airways, and accounts for about 5% of the deaths worldwide, and then is one of the leading causes of death globally. There is no existing drugs. The treatment is only to reduce risk of death. We now have a lead candidate which has been developed according to Respiratorius strategy, focusing on small and human airways. And it is a new chemical entity with both anti-inflammatory and bronchodilatory properties, equivalent of those the RESP-1000 project, which is more or less similar to 9000, although it is completely outside the previous patents from the 1000 project. Our focus has always been, as I said, to use human lung tissue in our development, and we have been able to have a set of lung tissue from about 100 individuals, and our compounds have shown significant and consistent bronchodilatory properties. For the new candidate drug, we have positive preclinical efficacy and pharmacokinetic data, and we have also performed an in vivo study showing significant and consistent bronchodilatory properties. During the last couple of months, the project has progressed tremendously. We have initial safety studies which has been done according to non-GLP, where we see that we have no significant deviations from at the concentration considered relevant. And those studies will be followed up according to GLP, both in vitro and in vivo. We have also performed a pilot study in ex vivo study that is similar to those experiments that has been done before internally, but now we have performed at an external CRO, And we see that the new candidate drug has expected efficacy and similar to previous trials. The next step will be to expand the study with tissue from other species and of course, most important, from humans. The production process of the new chemical entity or the lead candidate drug is established, and we are now moving into GMP production. We are also setting up a collaboration with a specialist in inhaled medicines, being responsible for the formulation of the new drug. The overall value enhancing milestones for the years to come in order to take the asset into a phase 1 study is of course the application to start the phase 1 study. And in order to file the application, we need to first of all, we need to initiate and conclude the toxicology study, and we also need to compile the regulatory documentation of the new substance, which I reported on previously, which is advancing, and we need also to conclude and finalize the GMP production for the clinical studies. And we also need to have an optimal formulation of the new inhaled drug and production of the substance. We have a preliminary examination of our patent application, which is a new chemical entity, and that is now being forwarded to the different countries where we have applied for a patent. And also for the financial part of it, we will have a cash infusion from exercise of warrants, which is in the range of 19 to 25 million pre-cost, and will be on our account in Q3 2021. And that concludes my presentation of Respiratorius.
Thank you so much, Johan. Let's move on to some questions then.
Sure.
The biggest question for the year, I guess, is whether or not you will find a partner for VL001. And have you had any interest? Can you tell us anything about the interest?
Yes, we have interest, and we have a data room, and we have activity in the data room. But as I presented the value of VL001, we see a great potential in the project. So it is not just an agreement. It is also to have an agreement that we foresee is similar to the value we actually see of the project. So yes, we have had interest, and we also have turned down a couple of offers.
And then moving on to looking more at the patent situation, what does that look like for your two main projects?
I would say it looks very good. First of all, we have a patent granted in the most important market for VL001 for the overall concept. And then we also decided, although we have both the patent and we have also the orphan drug designation, we decided to also file a patent in the early stage where we on the formulation of VL001, and that was done actually before we entered into the development. And since then we have progressed in the development, we have learned a lot, and we have a new concept of the formulation. So yes, we have hopes on the original application will be granted, but we can also foresee that we will follow up with new patent application as well. And for the RESP 9000, we have a positive examination, and we have now forwarded to the different countries. So we have hopes for the first granted patent in that family. I cannot say when, because I don't know, but still it looks very good, I would say.
So if we then turn more to the financial side, like you mentioned here, you have warrants.
Yeah, we have warrants, and we just recently finalized a issue of shares. And that issue of shares is those assets are directly directed to the PK study and finances the PK study and production of the VL001 for the PK study. And the warrants that will be exercised mid this year will be directed to both the toxicology study and concluding the preclinical research of the RESP-9000.
Well, it sounds like you have a lot of interesting things coming up.
We have a lot on our plate, yes, and we have made great progress during the last couple of months, and much accounting to that we have an organization now in place also.
Well, we look forward to following you during the rest of the year, and thank you for coming.
Thank you very much.
And then I am joined in the studio by Morten Albertsen, the CEO of FluoGuide. Welcome, Morten, and please go ahead with the presentation.
Thank you very much for the introduction, and welcome today. I'm going to present FluoGuide, and FluoGuide we maximize surgical outcome. We do that by intelligent targeting, and the first area we start out with is oncology surgeries, because it's a quite, first we need the disclosure. It's quite a big market, and every year 15 million patients will be diagnosed with cancer, and about 80% of them need surgery, sometimes during their disease. And half of them will have recur of the cancer after surgery. And that's not because the surgeon is poor in any way, it's simply because it's very hard to see where the cancer stops and where the normal tissues start. And our solution to that is a simple one. We have an UPAR targeted illumination of the cancer, so the surgeon can see all the cancer and remove all of it the first time. Our lead product, FG1, has been undergoing clinical testing, and it's been demonstrated to be well tolerated, and also that it lights up cancer in humans. We have a strong foundation, we have a strong IP on UPAR targeted surgery. We have a good cash position. We are listed on the First North with a market cap on approximately 1 billion, 1.2 billion, 1.1, 1.2 billion SEK. And then we have an experienced team and quite broad expertise from the very early stage of discovery up to commercialization. And our procedures is a very simple one, and yet it has a quite profound impact. This is one of the things that is very important with FluoGuide, that everybody likes us. We have injected our product just prior to surgery. It will circulate in the body. It will bind to the cells expressing the UPAR, and that's cancer cells. And during surgery, the surgeon will switch on the light, and they can see the cancer and they can remove all of it. I mean, the patient obviously have a benefit from this because they can be cured fully the first time, but also the hospital can save a cost in more aggressive treatment, repeated surgery, and the surgeon becomes better to do what they really would like to do, namely cure the patients. Last but not least, the equipment manufacturers also benefit from this, because what we can do is that we can get extra features of the equipment in play, and that means that the equipment manufacturers either can charge a higher price for the equipment, or they can get a better penetration, and that's simply because we help the equipment manufacturers to add more benefit from the equipment. So this is one example of a study we did. It is in mice, but it's a human surgeon, it's a human equipment, and it's human cancer. We took the surgeon and we asked him to remove all the cancer he could under white light, and there was 8 animals in total, and here he removed more cancer in 4 of the animals. Then we switched on the fluorescent light and asked if we could see more cancer, and he wanted to remove more cancer, and he did. And he removed more cancer in 4 more of the animals. And it was of course all validated by pathologists that it was cancer he saw. So that means had it been patient, had the last 4 patients had gone home and actually come back with some aggressive or some developed disease, or even have been died in the end. So this is what we are doing. One thing that's quite important is that we use standard equipment. We want to use only standard equipment, so we have a plug-and-play situation, and we don't want to sell equipment. We really want to just fit into the current equipment in the surgical rooms. So UPAR is our target, and it's an enzyme system used by cancer cell. When it's spread into the normal tissue, it breaks down the normal tissue to get in, and that is really what the UPAR is doing. So the more aggressive the cancer is, the more the spread, the more UPAR is expressed. And you can see on the right side of the chart here, you can see that some of the cancers are of course more prevalent. It could be, for instance, colorectal cancer, lung cancer, and breast cancer, and some of them are more severe, but with orphan drug designation. Our first indication is glioblastoma, and the reason for that, there's a couple of reasons for it, but one of the reasons is that there's a huge unmet medical need for glioblastoma. Only one patient out of 20 will live in 5 years, and there's no surgeon are very used to use equipment, so that is another aspect of it. The second thing that is very important with FluoGuide and make it very interesting as investment case is that we have a very short path from where we are now and until we get to a commercial product. We have only 2 studies that is needed to be done, and the patient is included shortly into the study. So that means, as I shown before with the animal experiments, the patient are included. They have done, the surgery is done under white light, the fluorescent light is switched on, and then what lights up is, is sent to a pathologist to validate that what lights up is cancer and what does not light up is actually normal tissue, and then we have the results. So we need a safety study first, and then we need a confirmatory study second, and then we could have a registration in 24. The first study is the one we have currently ongoing, and it's divided into 2 phases. The first phase we're looking primarily on safety, but also we get some efficacy readout. We have a dose selection, and then when we have that, we go into the second phase, and that is basically a mini phase 3 study. So we do exactly what we need to do in the phase 3 study, just in a smaller scale. And right now we have a very good indication that the product FG1 works. It lights up cancer, and we know it's well tolerated in patients. We have 5 cohorts, so that means that we have 5 groups of patients with different doses that we continue increase until we get no better imaging effect. As you can see, that actually there's 16 patients, not 15 patients. That was because one of the patients had a non-derived, brain-derived cancer, and we actually also demonstrated that FG1 works there. But that happens sometimes from the neurosurgeon, that they include a patient that is not with glioblastoma, and that's what happened in that single case. We have the data will all come out this year. We have the safety data in Q3, and we have the final result in Q4. It's very important that we can already now see that the cancer lights up, and that means macroscopically we can see what is cancer that lights up. But what really is interesting to see what benefit we can really provide to the patient is to understand from the pathologist if the small island of cancer, we can also detect those, and if we can see the cancer in depth, and that result will come with histological examination done by the pathologist. And we have the first preliminary ones done in Q3 with the first part, and then we have the final ones in Q4 with the second part. Our pipeline is that we have glioblastoma as a first indication. Then we want to we just raised money now here, so we can actually go into a more prevalent indication, most likely lung or breast. And then we have FG2 that we licensed in last year, and have some different features that it is treated differently from the body and works on some different cancers. For instance, colorectal cancer would be better suited for FG2. And then last, we just in-licensed the use of photothermal therapy, which really is a technique where we can destroy the cells that FG1 is bound to. So by lighting at it, we heat it up, and it will be destroying just the cells around FG1, but not the tissue on top of it or beside of it. That's a very neat feature, and really translates us into treating and helping the surgeon to treat the cancer, not only guiding them. And we, of course, have very good data on that, and that will be going into clinical testing next year. So the competitive landscape, I mean, 2 products have been approved now in the landscape, and they are on the down and on the left side. They are very cancer indication specific, but they're not cancer specific. So that means that they are converted to the fluorescent product inside cancer cells, and then they kind of float around the cancer and form a kind of cloud around them. They can only be used in one indication, it's glioblastoma and bladder cancer, respectively. And more importantly, they are using visible light, meaning that you can only see it on the surface 1 to 2 millimeters into the tissue. You cannot see it into the depth of the tissue. All the other products up there on the chart, they are based on something that targets specifically to a receptor, and they're based on near-infrared light that you can see 1 to 2 centimeters into the tissue. Some of them are equipment dependent, and for FluoGuide, where we're focusing on UPAR, we have this unique feature that we on the same time are very cancer specific, so we only bind to the cancer cells and then we are cancer type unspecific, meaning that we actually can go in the glioblastoma, and then we can spread out to the more prevalent cancers with the same product. And that is a third thing that is very important with FluoGuide. We are riding on a very nice trend in the surgical scene. What happened is that a lot of technology development is coming into the hand of the surgeon, and that makes them better to do the surgery. And there could, for instance, be microscope for the brain cancer, it could be scopes for the lung or for the stomach. And as I mentioned before, we make them all better to do what they already do. They need really something to guide them. It's not only FluoGuide, it's all the present guided companies. But really what it means is that there's enormous synergy between us, and that means that the manufacturers really want to see us succeed and help us move forward. And that is really very, very nice. The team behind, we have Andreas Kjær, that is the scientific founder of the company. He's doing the research, have a group of 50 people on the Copenhagen University. He's a board-certified nuclear medicine chief officer, doctor, and he's working with cancer imaging and diagnostics. Then Grete takes over and is doing all the production and the preclinical testing. Dorte takes over, then doing the clinical testing, and she both have the expertise within devices and drug, which is quite important. We are under the drug regulation, but a lot of philosophy is device. Then Henrik came aboard as CFO last year, and it's because we are doing more commercialization now, focusing on that. And he also is one of the first, founder of one of the very first companies in Denmark back 30 years ago almost. Myself, a medical doctor, commercial, and have done this before with another company, Novi, taking a product from the bench to the market within oncology, and now it's FluoGuide's turn. So we just announced very recently that we had done a directed issue, and that was primarily to get more institutional investors on board. So we now have approximately 23% of our shareholders are institutionals. We have more than 9,500 shareholders in total. And we have had a quite nice development. We was awarded as the best IPO in 2019. We have European funding of 2.5 million euro. Half of it has been paid out, other half this year. And then we have a strong incentivized management and good two supporting investors, Link AB, and also a Danish mid-size bank, Arbejdernes Landsbank. So in short, there's three things that's important with FluoGuide. We address a huge market. Many patients get cancer and undergo surgery. We have a unique technology, the UPAR targeted illumination of it. We ride on very nice trends, digitalization, minimalization, and automatization of surgery. And then we have shown with our first product that it works and that actually it's well tolerated. We have many and important milestones coming up over the next period of time. Safety in Q3, efficacy in Q4. We start the prevalent indication study in Q4 as well, or Q3, Q4. And then we will have some very important milestones next year, transforming FluoGuide into a late-stage clinical stage company, starting our phase 3 study, start clinical development of our second product, and also developing the photothermal therapy. So quite exciting time for FluoGuide patients and shareholders. Thank you.
Thank you so much, Morten, for that presentation. I was wondering, what kind of feedback have you had from the medical profession about your product?
Mm-hmm. Well, I mean, it's a very good feedback actually. I can give just a small story. I mean, we have had through this corona a lot of clinical trials have been stopped or been put on hold. And we have an extremely dedicated neurosurgeon. So she's kept going. Of course, glioblastoma is a very severe indication, and therefore patients with glioblastoma will get into the hospital to have surgery taking place. But still, there was a lot of the studies that have been locked down in the hospital. Our kept going on a recruitment rate that we actually anticipated. She had an accident with her foot, or sometimes she went into the surgical room with a chair and just did the surgery anyway because she's so eager to get going. So we have very, very good interest. And what we have seen during the study with this accidentally included patient, that actually she have a new indication she would like to develop, and then she have a third one also when she just, so she's quite intrigued by this, I must say.
We turned looking a bit more at the business strategy of things. In February, you changed your listing to Nasdaq First North. Why did you make that move at that time?
Yeah, I mean, we kind of, before Christmas, we reached an important milestone. We have shown that our first product now work and it was well tolerated in the first cohort that were treated. And the next phase of FluoGuide really is a more international. We have to go to key opinion leaders, we have to go to regulatory agencies, international partners, investors, and many of those are not in Denmark or Sweden necessarily, so we would like to be in a more recognized marketplace. Another aspect of it is that it's important for us long term to have some in a base of institutional investors as well, to provide robustness and validation, but also flexibility when we do the partnering, that they know that there are an alternative for us really, which is very important for us to make good deals. So for us it was quite important to move to the Nasdaq to prepare that, and actually this directed issue we just did here was actually in the plan to strengthen us and make it more likely that we get to the market.
And if we finish by looking ahead a little bit, what news can we expect from you for the rest of this year?
Yeah, well, there'll be many exciting news, but I think some of the key things on the slide here, really safety in Q3, and with the safety in Q3, then we are over this safety part of the study. So that means that the safety part of the study is done on a very controlled conditions, and as soon as we're out of that, we have a license to hunt other indications more freely, and that's when we will start looking into other indications. So that's a very important milestone. Then we will file for a couple of clinical trials in more prevalent indications. That's very important. And for this one, we get the first indication quite early on, because we can only half blind it because the surgeon can see it. So that's a good sense, in the sense that we get a quick result. We have to treat the patient very shortly, so the study we're doing is very short. So, well, safety, efficacy, benefit, regulatory interactions, that's what you can expect. So a lot of exciting things.
Well, we will be looking forward to that, and thank you so much for coming.
Thank you for inviting me here.
Next up, we're happy to welcome the CEO of Glycorex Transplantation, Pontus Nobreus. Welcome, Pontus.
Thank you very much for having me. It's a pleasure to be here today to introduce Glycorex Transplantation. In the US and Europe, more than 120,000 people are waiting for a new kidney. And in the US, 20 persons a day is dying waiting for a new kidney. Besides all the medical challenges and the suffering for those people, it of course also creates a lot of cost for society in terms of dialysis treatment and other treatments. Glycorex is developing products that can safely and effectively contribute to reducing those waiting lines. And today I will talk about what we do in the transplantation area and how we can bring our technology into other segments as well. But let's start with the big picture. Chronic kidney diseases is a huge global problem. It's affecting 850 million people around the globe. And it's actually predicted to be the 5th leading cause of death by 2040. And by 2030, it's expected to be more than 5 million people receiving some sort of kidney therapy. And by that I mean either going through dialysis or a transplantation. Not only does this affect the individual, but it has also a huge impact on the society. As an example, in the US, the public healthcare provider Medicare spends about 7% of its annual budget on this 1% of the population affected. And the same goes for many, many countries around the world. So there is actually a big incentive to try to do as many kidney transplantations as possible, because not only does it provide better quality of life for the person and also socio-economic benefit, but also when you get a new kidney you have a longer lifetime compared being on dialysis. But of course there are some challenges when you do kidney transplantation. I already mentioned the gap between supply and demand. And there is another thing that we need to consider when we do transplantation, and that is the blood group compatibility. You can see on this slide in the middle, as you probably know, there are 4 different blood groups. And if you have the same blood group as the donor and the receiver, it's not a problem. But if not, you can have a rejection. So you have to consider that as well. If you do living donor kidney transplantation, there are also other ways to go around that, and that is by having kidney pad donation program. But that's also a little bit of challenge in many countries, because as an example, in some countries you run those programs maybe 3 times a year. You search in databases and you try to do this match. So if you don't have a match, you have to wait another 4 months. So that's not effective. And this is something that Glycorex is addressing. We have a product called Glycosorb ABO that can enable blood group incompatible transplantation. How does it work then? Well, if you consider the blood plasma in a normal person, you have about 3 liters of plasma. And in that plasma, you have a small amount of antibodies. It's about 1%. And what we do with our product is that we specifically remove those antibodies from the blood plasma. Without introducing any foreign material, we do it outside the body, and thereafter you can safely and effectively go ahead and do the transplantation even though you don't have matching blood groups. So how does it work then? Well, this is a product. It's called an immunoadsorption column. It's based on active carbohydrates. It's our own developed technology. And the nice thing with this medtech device is that it fits into standard plasma separation machines in the hospital. So there is actually no need for any extra equipment, no extra investment. A question I often get is, who is treated? Well, that's a very good question, and the answer is it's the patient that is receiving the kidney. And you are treated for about an hour, depending on the weight, and you can do that a couple of times. After each treatment, you measure the level of the antibodies to make sure that you have sufficient low levels of antibodies to be able to do the transplantation. So it's a very safe and easy method to prepare a patient for a blood group incompatible transplantation. And we have done this actually for 20 years. The first blood group incompatible transplantation with Glycorex was done in 2001 in Huddinge sjukhus. It was an 18-year-old woman who had been on dialysis since the age of 8 years. So suddenly, in a few hours, she got a new life. Since then, our technology and product has been used in more than 5,000 transplantations. And we have been focusing mainly on kidney transplantations and from living donors. But our technology has also been used successfully for heart transplant, liver transplant, and lung transplants. So the question is, how is the landscape in general around the world when it comes to transplantations? On this slide, you can have an overview of the development of the number of transplantations. And in 2019, there were more than 150,000 transplantations performed. And the majority of those were in the kidney area. And if you look in the kidney area, you can divide it into two pieces. One is kidneys from living donors, and one is from deceased donors. And the deceased donor area is a little bit bigger than the living donor area. And it has been a tradition that it has been a little bit of resistance to take a kidney from a healthy person. Even though we know that the outcome is better when you take a kidney from a healthy person versus from a deceased person. But that is often governed by national policies and regulations. But what has happened in the past years is that there has been a lot of lobbying going on to increase the number of living donor transplantations, given those long waiting times. If we break this down into geographies, we can see that the largest market for living donor kidney transplantation is by far India. And this is very interesting. In India, it's performed twice as many living donor kidney transplantations than in Europe as a whole. The US is also a large market, followed by Turkey and Mexico. And it's very positive that Glycorex is actually represented in 3 of the 4 largest living donor kidney transplantation markets. And we have ongoing plans for market introduction in the US. We have broken down those numbers into monetary terms. We have identified the market potential, and now we're talking about the living donor kidney transplantation market, which is just a sub-segment of the general transplantation market. And we have concluded that this market for our product that we have been using for 20 years is actually 600 million Swedish kronor. And the question is, how have we addressed this potential? Well, our main region is Europe. We have 80% of our sales in Europe. We have a strong market in Germany, our largest market. We have strong presence in Scandinavia and several other European countries. But still, we have more to do there. And our strategy in Europe has been so far to work with direct sales. And we do that for several reasons. 1, we get a higher margin. 2, we have better control of the sales process. And thirdly, we can increase our presence and interaction with the doctors directly. And that's get a better understanding of the business. And outside Europe, we actually work with distributors. And we are present in those, I mentioned those markets before, India, Mexico, and Turkey. India, we have been for a while. In Mexico, the first transplantation was done in 2018 with Glycorex, and we were introduced into the reimbursement system in 2020. In Turkey, we signed a distribution agreement last year, and this partner successfully took our product to the Ministry of Health approval in February this year. And we have activities underway in Turkey to get our product into the reimbursement system. That is, of course, a little bit longer process, but we also have sales activities because we can still sell the product. And the US is also a big market, and here we have an ongoing plan how to get into that market. So that was the transplantation market. The question is, how can our technology platform be used in other areas? Well, we have a very interesting area. Sorry, I'm now mixing the slides. I just want to mention one thing before about Germany. And this is a proof of concept about how we can increase the number of transplantation using glycosorb to a sustainable level. This illustrates that we can go up to 20% blood group incompatible transplantations by introducing glycosorb. And I'm convinced that we can take this concept to other countries with the right conditions and make a copy and paste. So that is very promising. So now I'm going to talk about other segments that are of interest for us. And we have blood plasma, which is a very important tool for healthcare around the world. I mean, if you look at it, massive bleeding is one of the leading causes of death. And if you identify all the injuries, 30–40% are caused by massive bleeding. So there is a need for plasma, but also a need for plasma for more of general surgeries, planned surgeries, childbirth, people suffering from anemia, people undergoing cancer treatments, and so on and so forth. And here it's critical to have a good timing and availability of the plasma, having the right plasma of the right blood group available at the right place. So that puts some challenges into the blood bank business. And here, glycosorb can help. If you remember on one of my previous slides, to the left here you can see how the matching when it comes to blood transfusion among blood group works. If you are blood group A and B, you can transfuse to all the other blood groups. But the challenge is that is only 5% of the population. So there is a limitation, and often you use AB plasma when you don't know the recipient's blood group or when you have an emergency. So what we have offered is a product based on glycosorb ABO. It's a new version where the blood bank, in an easy way, without any extra investment, can make their own universal plasma by using just gravity and no extra equipment. This is a totally different market than the transplantation market. It's a volume market, and we estimate the market to be about 1 billion Swedish kronor a year in Europe only. We are also very interested in another area that is affecting many people, and that is autoimmune diseases. And that is a very broad area. And we have narrowed that down in our work to a disease that affects up to 1% of the population in the world, and that is rheumatoid arthritis. And unfortunately, there is no cure to this disease. Of course, it can be in different levels and can go in cycles, but the fact is that up to 10% of the patients do not respond to the treatment. And we have developed a new version of glycosorb that we have successfully tested in biocompatibility studies, and our next step is actually to go out with a clinical study for RA patient, and if this is successful, of course we will then take it further to CE mark. I must point out here, this is a huge market, much bigger than the other markets for glycosorb. We mean that we have a potential of 5 billion Swedish kronor in Europe only for this product. We also have some other product in the pipeline, but now we focus on the RA product. So, to summarize, our main activities this year is to continue to grow the living donor kidney transplantation business. We have still many things to do in Europe. We need to focus on those large markets, India, Mexico, Turkey, the emerging markets, we are going to work with our registration work in the US. And for the UBP product, we actually had a CE mark last year and made a pre-launch where we went out to key reference customers for them to evaluate the product. Our aim is to have scientific publications and to use that as a sort of a platform for a more broader commercialization. And finally, like I mentioned, the RA product, to get that into clinical studies, given the COVID situation, of course, if we can do that. But that's our aim for this year. So thank you very much for your attention.
Thank you so much for your presentation, Pontus.
You're welcome.
Very interesting.
Yeah.
I just have a few questions for you.
Absolutely.
I was wondering what the situation is in terms of the registration process in the US, if you could go into that a little bit.
Absolutely. That's a very good question, of course. And when you look at the registration work in general in the US, I mean, there are different pathways and different tracks and different things you need to do. And of course, one very important thing is the study work, to present data, and especially data on patients in the US is very important for FDA. And what we did before the pandemic started, we had been in contact with hospitals that had an interest in doing such a study. And now, when the situation is improving, we can continue those discussions and go further in the planning of those studies. So that's one piece. Another piece is, of course, to prepare for the actual FDA registration work, to allocate the resources and identify the resources. So that is something that we do as well. So there are some parallel tracks going on there.
Mm-hmm.
Yeah.
Yeah, okay. Keeping busy there.
Yeah, absolutely, 100%. Yeah.
Well, I was also wanted to ask you about the competitive landscape for Glycorex. Could you go into that a little bit?
Absolutely. I mean, that is many different competitors in one way, you know, because in one way dialysis is a competition, you can actually say. But also there is used plasma exchange is also common, that you take, remember that I showed that slide with the 3 liters of plasma, that you take the whole plasma from the body and you put in completely new plasma. So that is used as well. And there are, of course, also other sort of, you know, columns that are used based on another technology. So there are, you know, in different ways, different sort of competitors, I would say, you know. Yeah.
Well, just to finish things off, I was wondering if you could tell us what milestones we should look forward to in 2021 from the company.
Yeah. I mean, what's very important for us, I think now when we have a new product, the UBP product that was CE marked last year, and we do all the commercialization work, we need to, you know, get publications, and we need to continue that work. So I think that is an important milestone. And also an important milestone is the RA clinical study, to get that going. And in addition, you know, to get going in especially, you know, these emerging markets when the pandemic situation allows that, because we have this huge potential in those markets. So these are, I would say, main things to look out for.
Yeah.
Yeah.
Well, thanks so much. We definitely look forward to continue following you, and especially with this RA project, seems very interesting.
Absolutely. Yeah. Thank you very much for having me.
Yeah, sure.
Okay, thank you.
The next company presenting here today will be Lipum, and with us we have Einar Pontén, who is the CEO of the company. Welcome, Einar, and I leave the word to you.
Thank you, Olivia. It's nice to be part of this Life Science Summit and have the opportunity to tell more about the progress in Lipum. And I have prepared some slides, surprisingly. Today I will take the opportunity to tell you on how we will exploit our lead candidate drug SOL-116, intended for treatment of chronic inflammatory diseases. Chronic inflammatory diseases affects millions of people worldwide, and inflammation is a natural process in the healing. And it is when this inflammation is lingering and becomes chronic, then the difficulties and the problems decreasing the life quality for the patients occur. And chronic inflammation is also a wide definition. Several hundreds of millions of people are affected by some kind of arthritis or inflammation in joints, affecting their normal life quality. But we have focused so far on the disease rheumatoid arthritis that affects around about 1% of the global population, and it's more common among women and girls. And the market for rheumatoid arthritis is around about 24 billion euro. The disease that affects children is called juvenile idiopathic arthritis, and that's a rare disease, decreasing the quality of life for children up to around about 16 years of age. Lipum is a biopharmaceutical company, and we are focused on chronic inflammatory diseases. We were listed on Nasdaq First North in April, just a month ago, and our lead candidate SOL-116 has been developed as a fully humanized monoclonal antibody, and is now rapidly approaching clinical studies. We have an experienced team on board, and the founders are still significant owners of the company, around about 18%. And everything originates from research at the Umeå University. We intend to develop something that is completely new and different. And this is based on the discovery of the molecule BSSL. It stands for bile salt stimulated lipase. It's a protein that we have demonstrated could stimulate inflammation. And for that reason, it's a unique target and will provide also a unique mode of action for treatment of chronic inflammatory diseases. This was actually discovered rather early, and that meant that a patent was applied for treatment of rheumatoid arthritis and juvenile arthritis that are granted in the US and Europe and some other markets. And the priority year is 2010, and it's still pending for the inflammatory bowel disease. We have shown in numerous preclinical studies, ex vivo and in animal models, that blocking BSSL has an impact on the arthritis. And in this study, this is called Pristin-induced arthritis model. We induced arthritis by Pristin, and on the control group, you could see that they developed a significant arthritis with a high scoring, while the animals that received an anti-BSSL antibody were significantly less affected. And there are two curves that are the field marks in the diagram, and that represent two different ways of administration of the antibody, subcutaneously and interperitoneal. A graph that like this illustrates with statistical significance the effect, but also you can very clearly see in the photos on the foot of these animals that the controls are significantly damaged, while the animal that received the antibody is in much better condition. So, based on this preclinical research, we have developed a monoclonal humanized antibody, and that is our lead candidate, SOL-116. It has been previously tested in at least 5 well-recognized animal models for arthritis, and based on that, we developed SOL-116 and have established a cell line. We have established a production method, and when this decision was made to finally elect SOL-116, we were able also to apply for patent for the product, and that was with a priority of 2020. So we expect to have possibility for patent protection until 2040. In addition to that, we also intend to apply for an orphan drug designation, and we see opportunities also to get additional protection by pediatric investigational plan and special exclusivity related to biologicals. Our clinical development plan, or our development plan for the company now, is focused on bringing SOL-116 to clinical trials. We have a production method, and now we are about to produce in large scale a quality, GMP quality of SOL-116, and that is ongoing. And in parallel, we have toxicological studies ongoing. We have just passed we are just in the middle of 4-week study in that program, and we will start a 13-week study after the summer. And altogether, then we will have the material for subcutaneous injection finalized beginning of next year. We will also have the toxicological studies finalized in the beginning of next year. That means that we can apply for clinical trial application and start the phase 1A study on healthy volunteers during next spring. And then we have in the phase 1 study, we have planned to run a phase 1B study, and that will be on healthy volunteers, adults having rheumatoid arthritis. And we wish then for results that we can both monitor the clinical effect by measuring biomarkers and by the traditional scoring made by the physicians. So this is sort of the industrial part of our development. Everything is planned and everything is progressing well at the moment. And in parallel to this, then, we have a lot of earlier preclinical results from more indications. We have reasons to believe that our target molecule, BSSL, has importance for various different chronic inflammatory diseases. And we have seen that there are elevated levels of BSSL in atherosclerotic plaque, and that is an inflammatory condition. We have seen elevated levels of BSSL also in patients having fatty liver. And we have also seen in studies on knockout mice that the absence of BSSL in a knockout mice has a protective power related to ulcerative colitis. But among all these indications we have studied most closely are, of course, rheumatoid arthritis and juvenile arthritis, where we have a comprehensive package of data that will enable us to submit a clinical trial application next spring. But during the upcoming years, we now focus on, in the preclinical work, we focus on studying more indications. So we have already started a plan for, well, we have an experimental plan for ulcerative colitis and IBD, and we will look into these selected indications more in detail, while the other program towards a clinical verification is moving on. So we have a two-step, parallel step strategy for the next years, and we aim then for a package of data consisting of the clinical verification in phase 1b study, that in combination with a package of preclinical data on more indications. And we think this will be attractive for partners, open opportunities for co-development, co-financing, getting access to a larger pharma companies, a network of clinics and patients, and knowledge about more indications. There is, of course, also an opportunity or possibility that we could run several phase 1b studies in parallel. So we are not waiting until end of 2023 to establish a relationship with potential partners. That is something that is ongoing already right now. At this point, there is also, of course, a potential for licensing, and there have been some quite interesting deals in the recent years for licensing with upfront money in the range of 70 to several hundreds of $1 million. And just recently, Merck also acquired Pandion Therapeutics, but that was an acquisition, and that was $1.8 billion US. And all these are in this autoimmune inflammatory field. So we are looking forward for value triggers. We have recently, of course, we should absolutely mention that it was a very important trigger when we could apply for patent for SOL-116, and that was made last summer, 2020. We have established a production method together with our partner Apcena in San Diego, and we have produced in 200-liter scale, and now we are preparing for 500-liter GMP manufacturing for the clinical program. And the toxicological and safety studies for SOL-116, they are just now ongoing by our partner Charles River, and so far everything looks good. We also have some collaborations, and one strategic collaboration is with the Örebro University, where we are involved in a long-term research project looking into the importance of interleukin-1, IL-1, but the project is broader, and our interest is in the field of atherosclerotic plaque that relates to heart attack and stroke, and in some way also to cancer. So that is something that was just recently started. And if we now have, looking forward, we have accumulated quite a lot of data that from the preclinical work and from the development of SOL-116, and we intend to publish this during the year in scientific journals. We will focus on more preclinical studies on more indications, and hope to be able to report data from these studies. And we will produce the GMP material for the clinical trial, and ultimately start then the phase 1 studies during spring 2022. I hope you had some interest in this, and we welcome you to search for more information on our homepage or on social media, or contact us directly. Thank you for your attention.
Thank you for your presentation, Einar, and I have some questions for you. Do you consider Lipum's drug candidate to be a game changer?
Yeah, we really do, because obviously with for example, rheumatoid arthritis, where you have a market worth around about 24 billion euro, there are, of course, a lot of drugs in use today, both small molecules and biologicals, and in fact also one of the most selling drugs are Humira, the most selling drug all times actually, and that is an anti-TNF antibody. It's something acting on TNF alpha, and these drugs are dominating, but they are also well known to have problems with non-responders. Around about 30% of the patients are non-responders, and approximately around about 30% also stopped to use the drug after some year because of decreasing effect and also because of side effects. And there are also JAK inhibitors and some other drugs that are used and as introduced, but there are few really new alternatives presented that we are aware of. And BSSL is sort of what you could say a newcomer to the field that has been overlooked by other companies, and we think our clinical data demonstrates clearly that this could have a very interesting mode of action upstream all other drugs that are used today. And also we have data on knockout mice, the mice living without the BSSL molecule, and what we can see is that they are living just as long and are just as healthy as the wild type mice. So we have reasons to believe that blocking BSSL with an antibody could be much less negative for the immune system, with less suppression of the immune system, and that could also correlate then to less side effects. So yes, we think SOL-116 could be a game changer in treatment of chronic inflammatory diseases.
And can you tell us a bit about the research that led to Lipum's founding?
I like the story myself because it's what I call an orthogonal discovery. It was in Umeå, in the academic research, the founders of Lipum were in the field of pediatrics and were studying the nutrition of newborn children in breastfeeding, and then BSSL was discovered as an enzyme important for the fat absorption by the infant. But much later, when they were studying BSSL molecule, they discovered it was also present in the circulation. And when they were looking into this more in detail, they discovered that BSSL was at elevated levels in places where you could find inflammation, for instance, in fatty liver and in sclerotic plaque. And that was the reason why the founders and the researchers decided to look into this more in detail, and could then later on verify in numerous of animal models that BSSL plays a role in inflammation, just as I showed you before.
Very interesting presentation, and thank you for joining us today, Einar.
Thanks a lot, my pleasure.
Next up, we have the CEO of Initiator Pharma, Klaus Elsborg. The company is developing drugs against erectile dysfunction as well as other indications. Welcome, Klaus.
Thank you very much. Yeah, today I'm going to present Initiator Pharma and some of the things that we do. So Initiator Pharma is a safe neurotransmitter modulation company. We focus on central nervous diseases where there's a large unmet medical need, and we go in and we modulate with our compounds the monoamines in parts of the brain where it's needed. So we have two programs in clinical development, IPET 2015 and IP 2018, for erectile dysfunctions, for the non-responders to the current marketed treatment. We also now have added a new program to our clinical pipeline in neuropathic pain, more specifically trigeminal neuralgia. This program is called IPTN21, and it's also the same active pharmaceutical ingredient as we see in the IPET 2015 program, and I will tell you of the benefits of that later. So what is monoamines? Monoamines are really important signal molecules in the brain, in a part of the brain where we have these monomeric neurons that actually are sending signals by transmitting it through these molecules. Some of the most classical monoamines are dopamine, serotonin, norepinephrine, and these are involved in a number of phenotypes and physiological relevant functions, such as emotion, arousal, and some sorts of memory. Also, it's really important to understand that one monoamine is not reliable for one thing, so you cannot just increase the level of one monoamine and then get one specific response. It's the level of monoamines that are important. So when you modulate the monomeric system, you need to do it in a balanced and a safe way. And this is what we do in Initiator Pharma with a new generation of proprietary drug molecules. Also, what's important to understand when we talk about the monomeric system is that this is actually a truly validated pathway for treating many different indications. There's been drugs on the market targeting various diseases through the monomeric system for a number of years. So not alone are we coming into a field where we want to treat patients, it is also a validated pathway. So we know that the mechanism works for treating a number of diseases. So what is the unique offering of Initiator Pharma? In Initiator Pharma, we have de-risked clinical assets. We have assets that already have been in patients, so we already have seen that they are safe. We have a highly experienced development team behind it who's been developing drugs for many years, and also who is competent in the many different levels of drug development. So we have the clinical trials that we are working with are relatively short. So with a modest investment, we can actually reach multiple inflection points in these trials. And also we can see that the efficacy endpoints in the trials that we are working with are actually very well validated. So it means that we don't need to invent something new. It's actually something that's already established with the regulatory authorities. We also see that as a very interesting and very advantageous risk-reward profile. As you can say again, we are working with a validated pathway, so we know what are the upsides and the downside of working with the monomeric system. And also we can see there is a potential to, now with the neuropathic pain, go into an orphan drug indication where we can get all the benefits of working with an orphan drug. So we are dedicated to developing novel and safe CNS targeting drugs. We still have full priority on our three clinical programs. So we have a clinical program for organic erectile dysfunction in partnership with MAC. We have filed the clinical trial application and we are waiting for the approval. We have the 2018 program in psychogenic in an ongoing phase 2A. And then we are planning now to do the trigeminal neuralgia, starting out with a proof of principle trial in trigeminal neuralgia. So we are actually again saying we are working with full focus on all three clinical programs, and again we are targeting a validated pathway with proven efficacy. So what is it we are targeting? What is the market that we are targeting with the various programs we have? We know that more than 300 million men are suffering from erectile dysfunction, and about 30 to 40% of these men do not respond to standard of care. These groups can be split into two groups, what we call organic origin ED or psychogenic origin ED. We estimate that there could be a potential annual sales of $1 to $2.3 billion per year within the organic non-responders to the PDE5 inhibitors based on a premium pricing calculation. And for the psychogenic origin of ED, we anticipate as annual sales of $700 million to $1.9 billion per year. In the trigeminal neuralgia, which is an orphan drug indication, so that's a lot less patients than we see in the ED, We still estimate with the premium pricing we can get for such a drug will be between $500 million up to $1.4 billion. So it's very, very high value and very diversified markets that we are targeting with the clinical programs. And by having these 3 programs, we actually have multiple shots on goal to make it a commercial success. So why invest in Initiator Pharma now? Because right now we are on the brink of starting delivering a lot of true value inflection points in the company. So as I said, we have filed the phase 2A application. We expect to get the approval, during the next couple of months, so we can actually start up the phase 2B trial in erectile dysfunction, in organic erectile dysfunction, in Q3 of this year, depending on the COVID-19, of course. Also, we will get the completion of the phase 2A in IPED 2018, the psychogenic erectile dysfunction. Those data are coming out. And also we will be able to initiate the proof of principle in pain study in healthy volunteers, which is part of the development for the neuropathic pain program. In the subsequent year, in 2022, we'll have the completion of the phase 2B study in organic erectile dysfunction. We have the completion of the proof of principle in pain, and also we'll have the completion of a MAD study for the IPTN 2021 compound, which is the same compound as we use in IPED 2015. And then, of course, we will have outlined the complete regulatory strategy for how to pursue the development into trigeminal neuralgia and move as fast as possible towards a market registration of this drug. So who's behind the team? Who is it that runs Initiator Pharma? So today I would like to highlight a few of the people here. Magnus, the chairman, an MD from Sweden, has been involved in a multiple number of companies, is playing an active role in the board of Initiator Pharma, and has been a member and chairman of the board ever since the company's inception. On the development side, I would like to highlight 2 people today, and that's Michael Thompson, the CDO of the company, who's the main driver behind all the clinical activities we have going on. He's the organizer, he's the guy who orchestrates all the different activities that we have, from the submission of the CTA filings all the way to conducting the clinical trials. He's getting good support from Allan Venert, who has more than 20 years of experience from Lundbeck as a VP, both in R&D and all the way to product launch with some of their products in the CNS area. So he's actually helping us, making sure that we are getting the right trial set up so we can get the right readout, so we actually have what the big pharma is looking for at the end when we need to go to the market. So right now we are actually announced that we had an ongoing financing of the company. We actually just got a 30 million SEK in a direct issue, and now we have a preferential rights issue ongoing for another 30 million. And actually this way we'll raise 60 million Swedish. There will actually be proceeds that will finance the total efforts of Initiator Pharma well into 2023. So we expect the preferential rights issue to happen during the summer. We need to write a prospectus, and we are waiting the feedback from the regulatory authorities there. But it's important to mention that it's a fully guaranteed preferential issues by the investors that will come to now. So we actually got a really, really strong syndicate of investors to support Initiator Pharma. We actually been working with large shareholder space ever since we started the company, and now we actually get 2 cornerstone investors to complement our retail investor community. We're getting Link in, which is a really strong investor in the life science field, and we're getting the institutional and Rodrigo coming in, supporting the company going forward. And we think this is, for the first time in Initiator, we have a good runway, so we can actually do the clinical development and the business development in the right manner. So this is really strong, and this looks good for the future of Initiator Pharma. So just expanding the pipeline, just a little bit on the pain that we are looking at. So neuropathic pain. So what we are looking at here, we know that monoamines play an important role in various forms of pain. We know that targeting monoamines, you can actually treat a lot of these pain symptoms. An example is the monoamine modulator called duloxetine, which was developed by Eli Lilly, sold for more than $4 billion at their last year of patent life. Is actually providing good pain relief in some indications. However, there's a lot of safety concerns with this compound as well. We see that there's an FDA black box warning on this compound, increased risk of suicide, sexual problems, and liver tox. With the IPTN 2021 program, we actually have a drug candidate that differentiates from duloxetine and will change the safety and efficacy paradox. So we are again developing proprietary and safe molecules to modulate the monoamine levels. Trigeminal neuralgia, the indication that we are looking to go in and position our drug for, is a rare disease. It's a pain, it's a stroke of pain going down through your face, down through your trigeminal nerve over your face. It comes like an extremely painful lightning bolt. It can come sometimes, it will last for 2 seconds. You can be hit by 20 or 100 attacks per day, or it can be a more general pain level all through the day. It can also often be provoked by touching, putting on makeup, and things like this. It's actually so painful that people often mention it as a suicidal pain. So it actually really, and also a lot of the patients are getting depressed simply by the risk of fearing the next attack. We see that it targets about 13 out of 100,000. We see it's twice as common in females above the age of 50. So you can split it up into 2 groups, the treatments that are there. You can either go in and you can do a surgery, and you can do that in the patients where you can see what is actually provoking the nerve. So if something is touching the nerve, a blood vessel or a tumor, those you can go in and do a procedure so you can kind of decompress the area of the nerve. Often we see that a lot of the patients that undergo these invasive surgeries actually, have a relapse of the pain coming back, and also there's a lot of side effects, numbness of the face and so on. But in the part of the patients where we cannot see the direct cause of the pain, the idiopathic patient segment are often treated with pharmacological therapy. And here the only FDA-approved drug is carbamazepine. Carbamazepine is an anticonvulsant that has a long list of side effects that is not desirable, but so far this is the only thing approved by the FDA. We can see that most of the patients are actually using other tablets on top of carbamazepine because it's not working efficiently enough. So there is a huge unmet need for new things, and we can see that our drug candidate fits very well into this space, and that's why we want to pursue this. So the way we want to develop this drug is we want to do a proof of principle study in healthy volunteers where we inflict pain, so then we can see that we have efficacy. We want to do that at various doses and with a benchmark. Then we will take it into a MAD study, so MAD study multiple dosing, where we'll actually be allowed to give it the compound for longer. So far we are only doing on-demand single dosing for ED. And then we will follow up with a patient trial in trigeminal neuralgia. This latter part is not fully fixed yet, but we have an ongoing discussion with the regular authorities how we can actually improve the trial design and get the best possible efficacy readout. And for this, we're actually collaborating with Frank Cesanowski in the US, who's actually a clinical expert with the FDA on understanding how to build and how to design trials in various indications. He's especially a specialist in orphan drug indications, and he's been working there for many, many years. And he also, out of the last 8 FDA accelerated approvals, he actually were leading 6 of them. So just briefly at the end on erectile dysfunction, which is still a big focus of Initiator Pharma. There's many, many patients here, and we see an increasing number of the patients do not respond to standard of care. So what is it, again, just to understand erectile dysfunction? We know erection is driven by stimulation. You see something nice, you're being touched, you remember something nice. And that actually leads to a release of dopamine in the brain. The dopamine is then driving through a process where you get a dilation of the arteries, getting increased blood flow to the penile tissue, and then you get the erection. Our compound is actually going and strengthening this by actually, making sure that the dopamine is present longer in the cleft between the neuron and the postsynaptic receptor, so we get an even stronger erection response upon stimulation. So what is it we're looking at the patients that do not respond? Well, we have a group of patients, about 30 to 40% that do not respond to the current treatment. We know in some segments, so for example, people suffering from diabetes, we know that this is even larger, up to 50% not responding to the current treatment. So for these, there is no treatment, and these are the patients we want to target with our 2 candidates, IPED 2015 and IPED 2018. So just briefly on the 2015, organic erectile dysfunction, you can say that's happening to us when we go older. The system gets less responsible to the stimulation. And also here you can see that this is simply by the system is not as responsive as it used to be. Here we are positioning 2015 as the drug candidate to go forward. We have done a phase 2A trial, where we included 12 patients, where we go in and we measure erection on these patients by having a retroscan on their penis, show them an erotic movie, and then we can measure the erection of the penis. So in this study, you can say it's a fairly small study, but actually what we measured here, that 25% of the patients actually respond to the visual stimulation. And this made us super excited, because here we actually can see for the first time that our drug actually can increase the erection in men suffering from erectile dysfunction that do not respond to the normal PD-5 inhibitors. Actually, the data was so good that we actually managed to construct a deal together with Mac, who's been doing the clinical trials for us. And actually they invested in the company, so they actually go in and they will take part of the clinical cost at running the cost at the Mac clinic, and that will be up to 23 million Swedish crowns, which they will convert into shares upon the completion of the trial at a 70% premium to the share price when we made the deal with them. So this is a true validation of our results. What we are doing now, we filed the phase 2B, so the phase 2B will include 120 patients. We filed that application with the regulatory authorities, and we'll come back to the market when that is approved and give more detail on the final design. But we are looking to, on the trajectory, to complete the active part of this study at the end of 2022, so next year. Briefly on the last project, the IPED 2018, the psychogenic erectile dysfunction. Again, here we're talking about a different form of erectile dysfunction. Here it's not so much being the age that's driving the ED, it's actually other causes like stress, depression, low self-esteem, or the use of antidepressants. So here you can say that even though it's ED at the end, it's very differentiated to the organic erectile dysfunction. Here we have an ongoing phase 2A study where we actually have a 3-way crossover at 2 dose levels in a phase 2A, same design as we did for the 2015 program, where we actually show an erotic movie and we measure the erection. And we expect to complete that study later this year. So multiple upcoming value inflection points, as I mentioned before, is that we're going to have the approval for the phase 2B protocol. We're going to start the phase 2B study. We're going to get the readout of the 2018 program in psychogenic ED, and then we're going to start the pain proof of principle study. So come join Initiator Pharma in our quest to make better and safer drugs for patients worldwide. Thank you.
Well, thank you so much for that presentation. First of all, it's very interesting what you're doing. I think the connection between erectile dysfunction and neuropathic pain, for example, that's an interesting connection, and depression as well. You mentioned that. But I wanted to start by asking you if you could guide our viewers through the most important milestones for the company in 2020 and also the beginning of 2021.
Yeah. So I think that, as I mentioned, I think that now with the we submitted the application for the phase 2B trial for organic erectile dysfunction for 2015. It's going to be huge getting that one started. We are really happy that we made a good collaboration with a partnership with Mac on this, so they actually take care of the cost of the clinical expenses, so whatever they do in their clinic. So this is really good for us, and we, that trial is going to read out end of next year, or we're going to finish the active dosing end of next year, and that's going to be, a life-changing moment for Initiator Pharma, because that's a true phase 2B with 120 patients included. On top of that, we have the readout of the psychogenic erectile dysfunction trial, a compound that I have very high hopes for. In the preclinical models, it was super strong efficacy with the 2018 compound, and we want to see this translate into efficacy in patients as well. And then with the new program that we brought in money for, the neuropathic pain, you can say that now suddenly we're changing away from being just an erectile dysfunction company. We're actually changing into becoming a CNS company focused on modulating the monoamines. And we know that the 2015 compound that we are using for trigeminal neuralgia is a super safe compound. It has a really good safety profile and has good efficacy in the preclinical models of pain. So we think that this is a good position to be in. We have one compound we know is safe, positioning for various indications, and see where we get the best results. So all this is really, really strong, and I look forward to the upcoming months and year here.
Mm-hmm. Yeah, well, it's I think it's interesting that you're going into more of a CNS indication, let's say, and I'm actually wondering, out of curiosity, do you see in the future going into other indications, CNS indications, for example?
Yeah, I think that we are always very opportunistic, so we have continuous looking out for projects that kind of matches the criteria that we want to bring in. We want something that is safe. That's the foremost and most important thing. So we actually like to look at programs that's already been in the clinic and that we have seen being dosed, like the 2018 program that came out of a phase 1 study from neurosurgeon Cereno back in the day. So we know that when we give the compound, we don't have the risk, we're trying to de-risk the program as much as possible by saying something that is safe. So I would definitely say that we are opportunistic on, going out and looking at other indications. There's multiple indications, as I said in the beginning, that where monoamines plays a really important role. And if we can see that there's the right play for some of our compounds or compounds out in the market, we will definitely consider that.
Mm-hmm. Well, going back to erectile dysfunction, that disorder in particular affects about 150 million men globally. And the figure is expected to increase twofold by 2025. Yeah. How does this affect the market value for your candidates?
I think it's, of course, this is really good for, you see an increasing number of patients in need. Mm-hmm. So you can say that we see that 30 to 40% of the existing population do not respond. But we know what's happening. People are getting older and people are also getting, more and more lifestyle diseases. So cardiovascular problems, overweight, and so on. And these are actually really important parameters when you look at the non-responders. Mm-hmm. So you can say that, so we anticipate that our share would be even larger. So more people will not respond to the PDE5 inhibitors, making even more attractive commercial opportunity for us. So we definitely see that. And we can also see in the literature now that people are actually staying active sexual for longer. Mm-hmm. So, before when you 10, 20, 15 years ago, when you turned 50, that was kind of the end. Now we see in Portugal, Denmark, Sweden, and the Benelux countries, we see people, very active up into the late 70s, even into the 80s. And of course, these men are going to have a greater chance of having ED. So that will definitely also increase the market in general.
Well, finally, last question. What will be your main focus for this year?
I think that the main focus is the clinical development that we are working on now. We are really focused on getting these trials conducted in the best possible way, making sure we maintain the high standard of getting the right patients in so we get a result that is meaningful. Just like we did with the phase 2A results coming out of the 2015, that is something that actually can facilitate that we can continue our discussions with Big Pharma and move the programs forward. Then, of course, we have a pipeline of other drug candidates in the discovery phase. We would still like to, spend a little bit of time on them, trying to see what we can use them for. And I think that also we will make some progress there in the coming year.
Mm-hmm. Yeah. Great. Well, thanks so much for joining us today and giving the presentation. And we look forward to following you in the coming months.
Thank you very much. And thank you for all the questions.
The next presenting company is Moberg Pharma, and with us today we have Anna Ljung, who is the CEO of the company. Welcome, Anna, and I leave the word to you.
Thank you, Olivia. Hi, everyone. I'm Anna Ljung. I'm the CEO of Moberg Pharma. We're a dermatology company that bases our products on proven compounds, which reduces time to market and development risk. And our lead program is MOB-15 within onychomycosis. And there we recently got phase 3 data on more than 800 patients, demonstrating that we have a world-leading ability to kill nail fungus. And based on this study, we're now aiming for EU submission during this fall, which would enable product launch in EU by 2023. And we're not planning this launch all by ourselves. We do have some good partners in place. So for instance, we have Bayer for the European Union, and we have Taisho for Japan. We also have Dongku, the market leader in dermatology in Korea, and Cypher for Canada. And in total, we're targeting category leadership with a product that we believe have $250 to $500 million annual sales potential. And in parallel with preparing the submission in EU, we're also preparing one additional US phase 3 study that will be needed for approval in the US. And that could also strengthen claims globally. And this is actually not the first time we're taking an onychomycosis product to market. A couple of years ago, we launched the first generation product, Kerasol Nail. And we actually built a 440 million franchise around that product. And we divested that 2 years ago. So basically what we want to do now is to repeat that journey that we did with Kerosol nail with MOB15 that has an even greater potential. And onychomycosis or nail fungus, is that really a big problem? Well, it's not life-threatening, but it's something that is very common. So 10% of everyone does have this, and it increases with age. So the elder you get, the more likely you are to end up with onychomycosis. And it's really difficult to get rid of that nail, fungi once it has gotten stuck under the nail. And there are really no good treatment alternatives out there today. So looking at the total population, well, there are about 30 million Americans that do have onychomycosis, but looking at the number of scripts, you only see 5 million scripts on an annual basis. So what that tells you is that there is really a lot of patients that do not seek treatment. And that is due to you have orals that are effective, but there you have the risk of side effects such as liver damage and also drug-drug interactions. And then you have topical treatments that are safe, but not very efficient. And looking at the data that we generated in this phase 3, in total more than 800 patients. In the North American study, we achieved 70% mycological cure, that is fungus-free. And in the European study, an even higher number, 84%. And this is world leading. It's on par with the oral terbinafine. It's the same molecule, but we're using it topically instead. And it's way better than any topical has achieved to date. So a really strong mycological cure. And normally, once you have achieved the mycological cure, then that is killing the fungi, then complete cure will follow. And that's really a combination of no visible fungi and the nail looks perfectly normal. And you see that for all of our competitors, there is a clear correlation between the killing of the fungi and achieving clinical cure and complete cure. However, what was surprising with our data was that we did have this very high mycological cure, but we didn't turn into a high complete cure. So we discussed with key opinion leaders and we spent significant time with the data. And it turned out that our vehicle, it is excellent in transporting terbinafine through the nail. It does so by increasing the water level in the nail. And when you do increase the water level in the nail, you get a temporary side effect, a temporary whitening of the nail. It's like when you have a long shower, then the tips of your fingers turn a bit whitish. This is the same thing for the nail, but you have treated the nail for a full year, providing a lot of water into the nail. So by giving the nail time to normalize the water level, then this discoloration disappears. So from a pharmacist, from a physician's perspective, you can't really say that the nail looks perfectly normal due to these whitening. But stopping treatment earlier enables more time for for the discoloration to disappear. So looking at the concentrations of terbinafine that we're actually getting through the nail, we get 40 times higher levels of terbinafine under the nail compared to oral treatment, and 1,000 times less in plasma. So we're basically taking the oral terbinafine that is gold standard and works, and we're applying it topically. We know that we get sufficient amounts of terbinafine into the nail where it's needed. And there is really no need for patients to use oral terbinafine and risk those side effects if there is an effective topical out there. So what we want to do in the next study, the study that is needed for the US study, for the US market, is to shorten the dosing regimen. Since we have so high levels of terbinafine, it will be sufficient to kill off the fungi with only 2 to 3 months of treatment. and we will have plenty of time for the water level in the nail to normalize until we look at the nails after 1 year, because you always need to look at the nails after 1 year, despite how long you treat, due to it takes time for healthy nail to regrow. So for a big toenail, it takes approximately a year for it to fully regrow. So having this shorter treatment time, it will, of course, significantly strengthen claims, not only in the US, but worldwide. And it's, from a patient perspective, it's, of course, a great improvement, since all the topicals are a 1-year treatment, and reducing that time, it's a big win from a patient perspective. And actually, looking at the patients, 3 out of 4 actually see improvement already at the first follow-on visit. So the patients can really see that something is happening with the nail and see it as positive. And looking at the way that our competitors, the prescriptions from our competitors, you have, based on the prescriptions, you see that average treatment time is roughly 2 to 3 months, although the prescriptions are really for 48 weeks, but no one really, the average consumer does not stand with a treatment that goes on for a full year. So great advantages compared to the topicals, and without the safety issues of the oral treatment. And looking at the market potential, well, out of these 250 to 500 million that I spoke about earlier, roughly half is in the US, and one fourth in the OTC markets, mainly Europe, and then 1/4 in other RX markets, such as Japan and Canada. And this is really driven by the difference between RX and OTC markets, is that the price levels are much higher in the RX, but of course volumes are higher in the OTC setting. And right now we have full focus on compiling our file for our EU submission. And there we have a dialogue with EMA on a pediatric plan. And that's really what is driving our timeline here. We have had good initial feedback, and we're expecting to get approval on our pediatric plan during the 3rd quarter. And that would enable us to file during the 2nd half of this year. And having this pediatric plan approved, that also means that we can go for a full application. That would give us a data exclusivity for up to 10 years after market approval, in addition to our existing patent protection. So we have patent protection to 2032 in all major territories. And provided that we submit during this autumn, we expect to get approval early 2023 and be able to launch in 2023. And in parallel, we want to do a meeting with FDA and get going with the US study. And as I mentioned initially, we do have some strong commercial partners in place. And the 2 most important ones, I think, is Bayer. That is the leader in OTC antifungal treatments with the brand Canesten, but they don't have anything against onychomycosis, so it's a really good match. And we also have Taisho in Japan, where Taisho will invest in 1 additional phase 3 study that is needed, a local study that is needed in Japan. And generally looking at this, it's the same model that we used when we launched Kerasal Nail, our 1st generation product, where we take responsibility for production and supply, and our partners take responsibility for marketing and sales. And that also means that the big investment in marketing and sales, that is carried out by our partners, not by us. So we're reducing the cash burden on a small company, and it also enables us to get the best possible launch for this product in all major territories. we actually decided very strategically to keep the US rights by ourselves. And that comes from not only US being the biggest market, so we want to secure our own destiny in the US, but also from our previous experience where we combined working with partners on a global scale and having direct sales in the US. And that was a really successful way to take this product to market. So in the US, and this was an OTC product, we actually had 30% market share. Now we want to do exactly the same thing, but with an RX product. And we think that we could be the market leader with an RX product as well. And as price levels are much higher, the potential is 5 to 10 times greater with the RX product compared to an OTC product. But with our OTC product, we did have all the big accounts, so Walmart, CVS, and so on. So it was actually sold on more than 30,000 stores. And that is, of course, possible with this kind of product. So even though we're a small team, working with the right partners enables us to also become a dominant player in this very niche area of onychomycosis in the US. And in addition to direct-to-consumer marketing, because that's really important in driving sales, also for an RX product, we will focus on podiatrists. That is a group that stands for roughly 40% of the US prescriptions. So it's a really important prescriber group for us. And it's a prescriber group that is a bit neglected, I would say, by the big pharma companies, because there are no really big products within this sector. So it's a good place to be if you want to build a small niche player in the US. And then we'll also, we're planning for a co-promotion with one of the big firms that has a strong sales force within dermatology, because that's the other big prescription group that needs to be covered. And to summarize, so with MOB-15, in these 2 large phase 3 studies, we have shown that we have world-leading ability to kill nail fungus. So 70 to 84% of patients become fungus-free. And we have the chance of becoming the global market leader within onychomycosis, and we see that the product potential of 250 to 500 million on an annual basis. We're now heading towards EU submission during this year, and that would enable a product launch in 2023 together with our partners. So we have Bayer, Taisho, Dongku, and Cipher as partners. So all the big territories excluding US, where we want to go ourselves. And in total, these partnerships, they enable milestones of $120 million in addition to revenue from product sales. And in addition to taking this product to market in EU, we're also doing this additional phase 3 study based on the learnings from our current phase 3s and reducing the treatment time, which we think will lead to even higher complete cure rates. And we could use that to strengthen claims globally. This is not the first time we're doing this. We do have a proven commercial track record from Kerasal Nail that we built into a 440 million Swedish kronas franchise. And we divested Kerasal Nail and that franchise 2 years ago for 1.4 billion Swedish kronas, and that was then distributed to our share owners. We kept what we think is the most valuable asset, MOB-15, next generation product that has an even stronger potential. So basically, we want to do exactly the same journey that we did with Kerasal Nail, now with MOB-15. Thank you.
Thank you for your presentation, Anna. And I have some questions for you. You will conduct another phase 3 study in the US for market registration. What will the study focus on, and what is the timetable?
Yeah, so the study will basically be very, very similar to the study that we have already conducted in the US. So it will be exactly the same setup. only shortening the treatment time. And by shortening the treatment time, we want to show that this discoloration that was seen in the previous phase 3, that will disappear once the nails have plenty of time for water content to normalize. So then we expect the same mycological cure, but we also expect a higher complete cure data. And time-wise, we're planning for this in parallel with EU submission and EU launch, of course.
And how would you describe the competitive situation for MOB-15, partly with regard to Kerosol nail?
Yeah, so the competitive position, it's really about, there are two alternatives on the market. It's either these topical treatment that is not very effective, or oral treatment that is effective, but you do have these side effect profiles. So there is really no great alternative out there today. So I think that MOB-15 has a very important role to fill, actually providing something that is both safe and effective to the patient.
And what would you say are the most important activities in 2021?
By far, I would say it's really getting this submission in place, because that will, of course, be the starting ground for our product launch in 2023. And also this, having a meeting with FDA, getting that next study going, because that will enable even stronger claims going forward, and also enabling us to launch also in the US, that is by far the biggest market for within onychomycosis.
Thank you for your presentation, Anna.
Thank you.
Now I'm joined in the studio by Axel Sjöblad, CEO of SenzaGen. Welcome, Axel, and please go ahead.
Thank you very much for having me. As you heard, my name is Axel Sjöblad and I am the CEO of the biotechnology company SenzaGen. Our vision is to replace animal testing with best-in-class in vitro technology, to establish a new industry standard and to contribute to safer products in society. SenzaGen was founded in 2010, was founded here in Lund, and it is a spin-off of the Department of Immunotechnology. We still have our headquarters here in Lund, and we also have a SenzaGen Inc. in the United States. We are listed on Nasdaq First North since 2017, and we are right now in a commercialization phase that started when I came on board in the fall of 2019. We aim at break even in 2022, and we are very happy to have a GLP certified laboratory since May 2020. Our technology GARD, Genomic Allergen Rapid Detection, is based on human cells, genomics and machine learning. It has the highest predictive performance of the tests currently on the market. Our aim, as you already heard, is to replace animal tests, and the areas that we currently focus on are to identify potential skin and respiratory sensitizers. Our business model, where we currently focus on Europe and North America, is based on sales driven from our headquarters. But those sales are also complemented with partner sales via licensed contract research organizations and distributors. The tests today we run at our laboratory, and it is also here in Lund that we do the product development. So, if we just take a quick look at allergies, which is a growing health concern in the world, we can state that up to 28% of adults and 23% of children in the general population are affected by allergic contact dermatitis. If we look at respiratory allergy, over 300 million people globally are affected by asthma. What you heard about allergy is also important input when we look at the market that we act in, which is the in vitro chemical safety testing market, which is a fast-growing market driven by multiple trends. The first trend is regulatory, where we see an increase of animal testing bans around the world. The second trend is ethical, and based on increased community engagement, both from individuals but also from organizations and companies in their work with CSR. The third trend is scientific, and based on a need for more accurate test results. And the fourth trend is economic, where there is a search for cost and time effectiveness in product development and also safer products for consumers. These different trends help us in the 4 industries that we address: pharma, chemical, medical device and cosmetics, and gives us a current addressable market of approximately 5 billion SEK. And that is a market that is growing with approximately 10% per year. So, what are we doing then when we talk about replacing animal testing with high-end technology? Well, we benchmark GARD against what we call traditional in vivo testing, which is symbolized here by the guinea pig and the mouse on the screen, which has an accuracy of 70 to 75% versus human data. And we also benchmark against the first generation in vitro, which has an accuracy of 75 to 80% and use single 1 or 2 biomarkers when it tries to identify the results. And against this we compete with GARD, which we call the next generation of in vitro testing. And thanks to the human cells, the genomics, the machine learning and an analysis of 200 biomarkers, we can offer 90 to 95% accuracy, best in class, and also potency information, which means that we can state if a sensitizer is weak or strong. So, having talked a little bit about the company and the technology, I would like to give you an update on where we are today. We are working, as I said before, towards a break-even target in 2022 and towards becoming a profitable, customer-centric company. We do this guided by 2 strategic goals: grow market presence and develop a world-class customer-centric organization, and 6 strategic initiatives, all aiming at strengthening our capabilities, processes, market presence and sales. So, let's take a look at these different initiatives. Under growing market presence, we have worked very hard during this pandemic time to intensify our market activities, the digital and virtual ones, with support from partners and different key opinion leaders. This has led to the fact that we have received important orders from both new and existing customers. Here I want to highlight a 0.6 million krona order for GARD Skin and GARD Air from a new European customer in the chemical industry, and also an initial grant from RIFIM, the Research Institute for Fragrance Materials, in the United States to test several substances from them with GARD Skin dose response. And in addition to these 2 orders, we have a steady flow of incoming smaller orders, which leads to a gradually growing revenue baseline. But we are not happy with that. After input from our customers, we have also decided to broaden our CRO offering. And we answer to customer requests by introducing new tests for skin toxicology endpoints that complement our GARD offering. And the first tests, new endpoints that we add are OECD-validated irritation and corrosion tests. And this expansion is part of our commercialization strategy and in line with our mission to offer the best alternatives to animal testing. The third initiative is about developing strategic partnerships. And here we have renewed and expanded our global collaboration agreement with the leading contract research organization, Charles River Laboratories. We have also expanded our collaboration with RIFIM in the US that I talked about before on the safe use of fragrances and GARD Skin dose response. And finally, we have entered into new preferred supplier agreements and master service agreements with global companies. And we have done that in the pharma industry, in the chemical industry and in the cosmetics industry. And it is from these customers that we have been advised to broaden our portfolio. That is why I talked about that earlier. Another important initiative under growing our market presence is of course for us to ensure regulatory acceptance for GARD. Here we work in line with 3 different streams. The first one is the inclusion of GARD in the update of the annex of the new ISO standard for medical devices. Here we have done what we can, so we are now waiting for the ISO organization to finalize its work. And we expect this to be completed in the fall of this year. The second stream is our MDDT submission to the FDA for GARD Skin medical device. Here we have replied to questions and provided the FDA with additional information. And here the aim is for the FDA to qualify GARD as a test for use in the development and evaluation of medical devices. And finally, which is very important for many of you that have followed SenzaGen through the years. We are waiting for the OECD adoption of GARD as official test guideline. Here, the EURL-ECVAM Scientific Advisory Committee, ESAC, is just about to finalize its evaluation of GARD. The next important step in the process is the experts group's report, which will serve as scientific evaluation and provide a clear indication of the group's stance on the GARD validation. The information we currently have is that the report will be released in June, so it is just around the corner. And we will of course press release the result of the report as soon as we receive it. And from then our objective is to refer to the ESAC opinion as soon as it becomes official in all the sales work that we do. Looking ahead, the next step is OECD's possible adoption of GARD as a test guideline. And here, to make sure that we don't lose momentum and keep the process going, we are in regular contact since long with both the Swedish Chemicals Agency and OECD. And we have already prepared the test guideline draft that they have seen, but we are of course waiting for the ESAC report to be able to include the correct final information in the draft. When it comes to our second strategic objective, develop a world-class customer-centric organization, I just want to highlight our continuous focus on sustainability. In this area, we drive the message that our tests contribute to safer and more sustainable products reaching the market, to a better work environment for the employees working in the companies in the different industries that we address, and obviously a reduction of animal testing. All of these different activities are fully in line with the UN Global Compact principles. So finally, if I can look ahead, I would like to say that we have managed quite well during this pandemic time, and we have had to be careful with the plans that we set in 2019 before the pandemic started. But we have managed wisely, and I would say that we are really ready to scale up as soon as the pandemic's short-term consequences are out of the way. And while we wait for the OECD validation, we will continue to approach customers that benefit from GARD's high productivity during product development and are interested in the additional skin toxicology services that we will be able to offer through our own GLP-certified laboratory. So with that, I would like to thank you for listening and open up for some questions.
Well, thank you, Axel, and I'm here with some questions then. Right now, many shareholders, I think, are interested in your sales development. What would you say about the current market situation?
Well, first of all, if I look back a little bit, of course we had to change the plans we set in 2019, as I think that most companies have had to do. But as I mentioned, we have managed wisely. We have focused very much on our own direct sales to have really the first contact with the customers ourselves, which has given us a lot of important input. And that has also given us access to more sales and also feedback from the customers. You know, I talked about broadening our test offering. So I would say that right now we have a steady flow of incoming orders, and we follow the plan that we have set.
And today most of the revenue is generated by your own sales team, but you are working on building a partner network. Can you tell us a little bit more about that?
Yes, the partner network is a very important part of our business model, but we realized during the pandemic that it was important for us to have this first-hand contact with the customers ourselves. So we decided to really drive that part of the business. But in the meantime, we have entertained the relations that we have with the partners and also added a few new ones, because when we move forward, they will become more and more important. And I think what is also important to highlight here is that our partners, they sell more of standard tests. So when we have the OECD validation or the ESAC report in place, it will be much easier for our partners to offer the GARD tests than it is today. You may also remember that I talked about that we have focused a lot on the product development phase with the companies and customers that we are working with, and that is easier for us to handle from the laboratory in Lund.
And finally, you mentioned that the aim is to break even in 2022.
Yeah.
In your opinion, what is the key factor for that to happen?
The key factor is now that the interest that we see for GARD, the increasing interest, continues to materialize in sales. That's the first and very most important thing. The second one is, of course, the ESAC opinion. If that comes out as we hope, I mean, it will be good support in our marketing and communication, and also for our partners that I just mentioned. And then as number 3, I think it is good that we also, you know, GARD is our spearhead product, but that we complement that offering with other tests that we bring into the laboratory. So we will continue, you know, to build on these 3 streams, and hopefully that will take us to the break even.
Yeah, well, we look forward to following you, and thank you so much for coming.
Thank you for having me.
Next up, we have the CEO of Cereno Scientific, Sten Sorensen. Welcome, Sten.
Thank you. So I'm very pleased to be here and present Cereno Scientific and our progress over the last period and our exciting future. So Cereno Scientific has a vision to transform treatments for common and cardiovascular disease. Cardiovascular disease is the #1 cause of death globally, and it's twice as many people die of cardiovascular disease than cancer. There is a significant market need due to the high mortality and the significant morbidity that is attached to this disease. Cereno Scientific was founded in 2012 out of research Sverkerjärn in Gothenburg. We have our headquarters in Gothenburg at AstraZeneca's BioVenture Hub, but our footprint has expanded significantly. So we have a daughter company, a subsidiary in Boston in the US, and we also have a significant research collaboration now established at the University of Michigan in US. We have a pipeline portfolio of a lead drug, a CS1, moving into phase 2 in rare disease and pulmonary arterial hypertension. And we have 2 preclinical programs in cardiovascular disease. Cereno was listed on the stock market Spotlight in 2016. Back to cardiovascular disease. It causes the most death worldwide, and about 22 million people are about to die of this disease by 2030. And as you can see on the right side here is significant cost to society, Europe illustrated here, and US. So there's lots of work to be done in cardiovascular disease to help these patients and all the stakeholders here. Many of the therapies are insufficient and can be improved, both for survival, better survival, and for better morbidity. We have our product portfolio in common and rare diseases. So CS1, as I mentioned, is about to enter phase 2 this fall. It's an HDAC inhibitor, and the indication that we are pursuing is pulmonary arterial hypertension, the rare disease. We have 2 preclinical programs, CS585, it's a prostacyclin analog, and CS014, it's also an HDAC inhibitor as the first compound is. CS585, we just acquired the rights to evaluate and then licensing in with the exclusive rights if the evaluation is positive. I'll come back to this later. Both of these preclinical programs are in cardiovascular disease. So if we go back here to CS1 and CS014 program, our clinical and one of our preclinical programs, they're both acting as epigenetic modulators. And epigenetic modulation is having an impact on gene expression without altering the structure of DNA. And our compounds are HDAC inhibitors known to be epigenetic modulators, and they've been applied for cancer treatment, but never, to our knowledge, for cardiovascular disease. And our vision is to unlock the potential of HDAC inhibition for cardiovascular disease therapies to the benefit of patients worldwide. And as I mentioned, CS1 about to move into phase 2 clinical trial for the rare disease pulmonary arterial hypertension. It is a rare disease that affects 5 to 15 people out of 100,000, and they experience shortness of breath, fatigue, etc. Initially leads to problems with oxygen supply and eventually fatal right heart failure. Lung transplantation is the only real cure, but most patients don't get that option. So there is a poor prognosis and unsatisfactory treatments for this disease. Now, why are we moving into pulmonary arterial hypertension with our lead candidate drug? Well, we have discovered that we have very beneficial effects mimicking almost the disease progression of pulmonary arterial disease itself. So we have documented antifibrotic, anti-inflammatory, pulmonary pressure reduction, and also antithrombotic impact with HDAC inhibition in our substance. So there's a very good opportunity here for us to have a positive impact for these patients. If we look at the market is around $7 billion today, and growing with to reach about 10 billion by 2027, when we estimate that we might be able to reach the market. So it's a significant market, it's growing, and the therapies there are insufficient for the patients. We applied to FDA and got orphan drug designation approved in March last year, and that gives us 7 years exclusivity upon approval on the market in the United States, which is the major market for PH. So what have we seen earlier with CS1? Well, as I mentioned, our preclinical and peer studies have shown that we have antithrombotic effects. We prevent thrombosis in vitro and in animals. We reduce pulmonary hypertension. We prevent fibrosis development and remodeling in rats with this pulmonary high pressure. And we also have anti-inflammatory and anti-fibrotic pulmonary vascular effects. So very interesting data. And we brought CS1 through a phase 1 trial with good safety and tolerability. And we also achieved our pharmacokinetics that we were looking for. In addition, we also saw a significant reduction of PI, a marker for thrombosis, and also for fibrosis development. So we have successfully completed the phase 1, and now we're preparing our IND submission for a phase 2, and that study to be started later this year. So the phase 2 study is about to start in September, and the principal investigator and the latest additional member to our scientific advisory board will be Raymond Benza. He's professor of medicine at Wexner Medical Center in Ohio State University, and he's one of the top thought leaders for PH in the world. We will look at safety and tolerability as the primary endpoint, but we'll also look at standard efficacy endpoint for this patient group. A calculated validated risk score will be measured, and we'll also look for efficacy and look for dose finding for the next study. We will apply cutting-edge technology for the pressure measurement. We will have an implantable device measuring 24/7 the pressure in the lungs. There will be 30 patients and about 6 centers in the US only. And the study will be conducted under the orphan drug designation status that was awarded by the US FDA in March last year. The expected timeline is start September and study results in the fall of 22. Very excited about this study. So moving back here to our preclinical development programs, and I'm very pleased to bring these up and present them because they are a result of our strategy to broaden our pipeline and build a biotech company in cardiovascular disease. So we have 2 programs, as I mentioned. CS585 just signed a deal and a press release about that deal this spring with University of Michigan. And since before, we have a CS014. CS014 is an HDAC inhibitor, and CS585 is a prostacyclin analog. And we just released a press release on our collaboration deal that we have made with the University of Michigan to study these 2 programs and bring those 2 programs and substances into man. 2 programs, 24-month each, and the programs will be led by Dr. Mike Hollenstadt. And actually this week we released a press release that we have signed and engaged Michael Hollenstadt as Director of Translational research for Cereno. So he will be in charge of these programs and lead them at University of Michigan, and also work with us in our senior management team. Just to highlight the impressive scientific advisory board that are backing our vision and our technology. We're Bertram Pitt as Professor Emeritus, University of Michigan. We have two, Deepak Bath and Gordon Williams from Harvard Medical School. We have Fayez Sanad from France. Gunnar Olsson, previously head of cardiovascular and GI research for the whole Astra Corporation. And the latest addition here, Raymond Benza, who is the top thought leader of PH globally and will be the principal investigator for our phase 2 program coming up. This is just to highlight our management team. We are 10 very ambitious, high energy people, and a lot of profession here, and believe that we have succeeded very well with our activities over the last period as the team has been built. I'm very proud of our achievements. As you can see here, we have new additions here, I should say. Michael Hollenstedt to the left in the corner, head of translational research. Tina Sepe just joined us as director for clinical research. She's in charge of the phase 2 program and the IND submission. And then also Stine Berg Hansen joined us as project director. So those are the latest additions. So we're a strong, competent team here. So to finalize, what has happened recently? We have strengthened our team for the clinical study, as I mentioned. We have expanded the team for preclinical phase with Mike Hollenstedt. We obtained actually 2 patent approvals in 2 key markets, Japan and Russia, this year for CS1. We in-licensed a preclinical program from University of Michigan, CS585, in cardiovascular disease, prostacyclin analog. And we have initiated our preclinical development programs for these 2, CS585 and CS014 at University of Michigan this year. Coming up, key milestones is phase 2 IND approval by the FDA, and then the start of the phase 2 program with CS1 in September of this year. So, just the last slide here, a broad overview of our strategy to build Cereno as a biotech company and provide value for patients and stakeholders, shareholders here. We start the phase 2 study in this year in September, and we have started our preclinical phase 2 programs at University of Michigan to bring those 2 compounds into man within 24 months. We expect to have our phase 2 program be able to report it the fall of next year, and we expect to be able to initiate a phase 2b/3 study in 2023. With regards to the preclinical phase, we expect to start phase 1 studies with CS585 and CS014 by 2023 in the fall. That's it. Thank you.
Well, thanks so much for that presentation, Sten.
You're welcome.
And I just have a few questions for you. You mentioned the addition of 2 new preclinical programs to your pipeline. I was wondering, how will those add value to Cereno Scientific in the long run?
Well, I think there are several aspects of why you build a portfolio and a pipeline if you want to build a biotech company. I think previously Cereno was a project company more than a biotech company. So we had one project moving CS1 into clinical phase. And now we have added 2 preclinical programs. So the reason that you do that is that you have the vision to bring new good pharmaceutical drugs to the market to help patients, but also you have a broader pipeline that has a difference in target, but also difference in timing. So you actually hedge your development programs when you have a portfolio, and you're developing them in parallel, but they have different phases. So it's the way it's done.
Yeah, of course. And actually, to add on to that, I also wanted to ask, both of these are through a collaboration with the University of Michigan. And how long do you think this, or where will this collaboration take you in the long run? Do you expect more preclinical programs through this collaboration, maybe?
Well, I should say that the current portfolio that Cereno has is pretty set now, just now, because we have our clinical program for CS1 coming up, phase 2. We have a CRO contracted. We're approaching FDA now for the approval, and we have the CRO to run the trial, etc., and the team to do it. And then for the phase 1 or the preclinical programs, we have that all agreed with the University of Michigan, and we have leadership in place and experience to get it done. So now we are very much in an execution phase of these 3 programs, the clinical and the 2 preclinical. But I would then again say that we are still looking for additions to our portfolio. And the addition of CS585, the prostacyclin analog, came about us being curious about what was there. So I think that was preparedness meets opportunity, so equals luck. But you know, it's good timing.
Yeah.
So I think it's not excluded that we will add other projects to, because we have a very strong cardiovascular team, both scientific advisory board and operationally.
Well, finally, last question. Of course, everybody's looking forward to the initiation of the phase 2 trial with CS1. But besides this great milestone for this year, what is maybe another milestone that we should be looking forward to from Cereno this year?
Well, I think that the start of the trial, obviously, but prior to the trial, we actually need the approval from FDA of the IND, investigational new drug applications. You have to get FDA's nod to go ahead and do clinical trials. So that application is going to go in, and then you get the approval, and we hope that we'll get the approval without too much discussions, and then we'll start the trial. The other thing that will happen this year is that although we did a directed share issue, a fast share issue in August-September last year that brought in around 60 million to the company, and most of that money is still in the company, I would say. But a financial event coming up in this Q3 is warrants package that was attached to that share issue. There was one coming up now in September and one next year. And those 2 warrant packages are fully prescribed around 100 million Swedish each. So those are financial milestones coming up, and they are important for us. And I think that based on our performance so far, people should be pretty interested to go into Cereno, I think.
Well, thanks a lot. That's very exciting times coming up for Cereno, indeed. And yeah, thanks again for joining us today.
Thank you.
Next up, we're happy to welcome Göran Konradsson, CEO of Ziccum, a company developing dry powder vaccines. Welcome, Göran.
Thank you very much, Michael. So what we do is that we formulate vaccines from liquid into dry powder that can be transported anywhere in the world to every child around the world. And this presentation, I'll explain what we do, and that is with the help of a movie. That's about 3.5 minutes. I'll explain also our business model, an expansion of our business model as I see it, but also about the market and all the players in this vaccine field that we see coming now more and more.
From the very beginning of human development, we've always sought to make products that are truly essential, as simple and easy to store as possible. And for centuries, that's meant drying them. Because when you remove water from a substance, you stabilize it, reducing the chance of spoilage and making it easier to control. Dried goods can travel further, last longer, and reach more people faster, more cost-effectively. Whether it's milk, rice, or medicine. So if we can do it with food and medicine, why can't we do it with vaccines? We all know that vaccines truly are one of our most essential health interventions, yet today, they're being held back. They require constant freezing or refrigeration, even down to -7 centigrade, specialist isothermal packaging, temperature tracking devices, fleets of refrigerated container trucks, specially adapted planes. All of it meaning that increasingly, vaccines just aren't reaching the children who need them the most. But there is a solution, and it takes us right back to air drying. Because now a new technology is for the first time air drying rather than freeze drying vaccines. That turns delicate liquid vaccines like this into thermostable vaccines that need no cold chain, like this. And vaccines formulated this way would finally meet the WHO's CTC vaccine requirements. They could be stored and transported anywhere in the world, reaching every last child in the world, with no refrigeration or freezing necessary. And what's more, they can also be produced at volume. Today, the vast majority of vaccine packaging facilities, known as fill and finish plants, use freeze drying, also known as lyophilization, to dry their vaccines. It's a complex, capital-intensive process. Even small lyophilization plants require large teams and energy-intensive vacuum freezing chambers, each of which need to be washed between batches with cubic meters of water for injection, an industrial solvent that also has to be produced in the same plant. All of which adds up to major costs, but in an aseptic air drying plant, those costs are halved. Half the number of staff are required. The central air drying unit, operating in an aseptic room within a room, is smaller, more compact, and requires far less energy than an industrial freeze dryer. And no water for injection is required at all. These savings mean that the actual plant itself can be agile, energy efficient, and compact. A modular air drying fill and finish plant would measure 36 by 27 meters and could be manufactured in less than 24 months on any greenfield site, anywhere in the world. Vaccines are not luxury items that should require specialist handling. They are true essentials. The technology to help reach every last child exists, right now. After all, you wouldn't dry your best linen by freezing it or baking it, so why do it with vaccines? With ambient air drying, we're no longer holding vaccines back. Robust thermostable vaccines need no cold chain and can build the lives and secure the futures of all children, wherever they are. Let's set vaccines free.
So coming back then to our business models. We started out as most biotech does in licensing, and since we have patent and we have new patent applications, and we have done an improved patent on our basic technology, and that was started out as being a basic model for licensing. But then we started to realize also if one would take our technology into a fill and finishing plant, what would that entail for anyone, any vaccine company? So we engaged another Swedish company called Key Plants, and they did a conceptual design for us, and then we started to realize, as you can see from the movie, that there was a significant savings in terms of cost and energy and also OPEX and CAPEX. So with that, we think there is an increased possibility to revenues and expand the worldwide capacity for fill and finishing, and this is a lot of discussion today these days about how to improve the world capacity in manufacturers of vaccines. But also it has to do with equitable access to vaccines. And temperature in the cold chain is one of the limiting factors that have been identified by organizations like WHO, and the difficulties in reaching out with the temperature sensitive vaccine to the children furthest away from the logistic nodes. So that's where we think we have a role to play. And in order to make this happen, there needs to be collaborations. And this has also been identified by many of the big NGOs and IGOs such as WHO and UNICEF, and we fully support that. And so we are in discussion with various countries, manufacturers obviously, but also NGOs and IGOs in order to make this happen. We've been working since more than a year with various types of contacts in these field in IGOs and NGOs. So we have regular discussions with UNICEF, the African Development Bank, and if you look on the NGOs pillar, we have discussions with CEPI, and there's a lot of things that's going on in this field, as well as, of course, with the vaccine companies. And we have been able to announce at least one collaboration with the name, and that's Janssen, which is an active collaboration that we have had since some time back, although it was somewhat delayed because of the COVID, but now we are collaborating with them. And that is one out of we have 3 industrial collaborations that we have. So from a point of view of Ziccum, what does success look for us? One is, of course, secure financing, particularly for the fill and finishing plant. Such a plant costs about $40 to $50 million to establish, and it takes about 2 years to do it, which is really very, very quick. Normal time, if you ask the vaccine company, it takes about 8 to 10 years to build a vaccine manufacturing plant. As I mentioned, access to bulk vaccine is key element here. We have always had a focus on children's vaccines. We think that's where the need are the greatest. But of course, with the COVID-19 and the new vaccines, the fantastic vaccines that have come, we're also looking at improving second generation or third generation of COVID-19 vaccines. Licenses is still an important part of our business models, and we are also reaching out to various countries for discussion about how can we help them and how can we help each other in order to establish a fill and finishing plant. And it has become very apparent and obvious now with the COVID-19 pandemic that many of the countries, for instance in Africa, they don't have any vaccine manufactured at all. In fact, only 1% of all the vaccines that is needed in Africa is being produced in Africa. So there is a lot of need and a lot of activities going on in order to secure supply and control over their own vaccines. And we hope and planning to see if we can play a vital role in this. So the concept we're working with is a module concept for manufacturing, developed then by Keyplants, and they have basically started up by containers. So you build up the vaccine plant here in Sweden, and you make it sure that it works, functions, you qualify the plant, and then it's being dismantled and it can be shipped to any green field in the world actually. And such a plant is not so big. I'll come back to some of the details about that. But here is the one example of drawing of such a plant, where we have ingoing material, ingoing people, and locker room for the personnel. We have the eye drying system, which you can see up in the left-hand corner, but also then the tableting machine, which is one of the bigger element in this. All of this is we're using standard available technologies. The only thing that's new is our own drying technologies, but all the rest are standard technologies for the industry. So some of the key numbers for modular plant for fill and finishing using our technologies, and as you can saw from the film, we reduce 80% of the energy because some of these lyophilizers are huge and requires enormous amount of energy in order to operate. And to do a lyophilization take 3, 4, 5 days. You reduce the temperature down to minus 70 or 80 degrees, and you have a certain cycles of this over the days. We have much less reduced OPEX, operational expenditure, as well as reduced CapEx when we compare with lyophilization. So the plant I just showed you has the capacity of 20 to 30 million vials per year plant, and you can have 1 or 10, or as you have in Pfizer BioNTech vaccine, or 5 or even 6 doses per vial. And with that, we can manufacture between 20 to 300 million doses per year. The footprint is 6 of a soccer field, and it's 1,000 square meters in 2 stories. The investment cost is around $40 to $50 million, and the estimated time for first plant when we start the whole procedures is about 2 years, a little bit more than 2 years. If you're more interested to read about it, we have some white papers on our website where you're more than willing to come and download the white paper and can read what we do and how we do it in more details. Very briefly about Ziccum, our company. It was established 2017 as a spin-out for Initiator Sciences in Stockholm. We went on the local stock market called Spotlight back in 2018. And as of 1st of December last year, we listed on NASDAQ First Growth. First North growth market, I should say. We have about 29 million Swedish kronor in our cash as at end of Q1 2021. We're about 10 to 15 employees. It depends a little bit how you calculate this, but it's including FTEs as well. As I mentioned, we have 3 industrial collaboration, where Janssen is one. We have an approved patent, which is quite unique, I think, for a small company like Ziccum. We have academic collaboration with the University of Marseille. They have a BSL-3 lab there and access to many vaccines and viruses around the world. So we have one system down there. And we are located in Lund in southern Sweden. And the management team that we have is myself, of course. We have Frida Jernberg, who's our CFO, but also, of course, Per Gärde, who's the inventor of laminar paste technology. So with those few words, I would like to end my presentation. Thank you very much.
Well, thank you so much for that presentation. I just have a few questions for you.
Yes.
First, obviously, the pandemic has put the spotlight on the problem of cold chain, having to keep vaccines in very cold temperatures. So have you noticed an increased interest from NGOs, for example, in states or also industry for your technology in the past year?
Yes, I mean, we've been very active, as I mentioned, to several of these organizations, so we have an ongoing discussion with them. It's a huge complex, obviously. There are many people involved, but there is a very nice, I think, momentum in the industry now to start changing things on how vaccines are being made and produced. So, yes, definitely, I would say.
Mm-hmm. And you discussed one of your corporate goals being plans for having a first dry powder vaccine plant or facility in Sweden. Mm-hmm. How are these plans progressing?
Very well, I would say. And we have drawn some of the facility where we are going to to place the plant, and we have a lot of good discussions around. But it's, as I mentioned, it's a big undertaking, of course, and many players needs to be in line before we are there. Having said that, there is a lot of initiative now amongst many of the organizations I just showed you in order to how can we increase the worldwide vaccine manufacturing capabilities, and we hope and we're striving to be part of that solution for the future.
Mm-hmm. And also you mentioned the expansion of vaccines to more difficult to reach areas. So what is the potential for your technology to gain acceptance, for example, on the African continent?
That's a very interesting question, I think. And as mentioned before, I mean, only 1% of the need of vaccines are being produced in Africa, on the African continent. And I think they had a bit of a hard wake-up call with the pandemic. There was a sort of an ice-cold shower for them to realize that, oh, nobody's going to help us. We're back in the line, last in the line, because of the rich world like us. We're living here in the Western world, which only constitute about 10% of the world population. So 90% of the world population, they don't have access to vaccine manufacturing plant where they could take control, or much more control over their own vaccine supply. So there is a lot of initiative now in the African Union, together with African Development Bank, for instance, that we know quite well. So yes, there is a lot of activities ongoing, because the situation that we have now with the pandemic, you know, it's not a sustainable way of doing things. So, and this has been going on for quite some while, I must say, because it's been the same situation with children's vaccine, but it hasn't been so high on the agenda. But now, finally, if I may say so, something good comes out of this is the realization that many countries need to take much more responsibility for their own vaccine supply, and not just depending on foreign countries to send vaccines to them.
Mm-hmm. Absolutely. Well, thanks so much for answering the questions and for your presentation, and we look forward to following Ziccum in the coming months.
Thank you very much.
Next up, we are joined via video link by the CEO of Gabather, Michael Robin Witt, who is joining us virtually from Södertälje. Welcome, Robin.
Thank you. So I'll go ahead with the slides that I have for this meeting. And we have a few questions, I believe, at the very end. So thank you very much for this opportunity to present Gabather. And I'll give you a snapshot of where we stand at Gabather now, and also a little bit looking ahead into the future, or what we have planned with Gabather for the next year or two with our projects. So Gabather comes from a combination of the words GABA and therapeutics. And GABA is the neurotransmitter system we work with, and therapeutics is what we want to develop targeting this neurotransmitter system. So what we're doing actually is developing new therapies against mental disorders in the general area of mental health. So one of the key things in Gabather with the projects that we work a lot with cognition. Cognition is a key feature in all mental disorders, and actually a key feature of healthy mental activity, and it's the way that we react to the world, the way we experience the world. And cognition is how we process the information that we get, both from an emotion and a reason point of view, and how we react to the world. So in the moment that this process is inhibited, or at least debilitated, like you can see in this slide, then we have a problem, and we cannot really react to the world in a proper way, because we experience the world in a way we cannot make sense of it. And this can be from a very slight, very annoying, perhaps, dysfunction, to very severe ones where we actually do not understand the world at all anymore. And that's actually a key feature in many issues of mental health and many issues in mental disorders, that we experience the world in such a way that we cannot understand it, and we can, the cognitive processes are deficit, and we react in it in a strange way, and that gives us reason to pathology. So it is the restating of the balance in the brain of the neurotransmitter systems I'm going to talk about later that we're working with at Gabather. So once you have functioning processing, both emotionally and from a cognitive point of view, you can react to the world in a proper way. So the cognitive effects are actually abundant in many mental disorders, many issues of health, mental health. This can be diseases from depression, schizophrenia, anxiety, bipolar disorders. All these pathologies have actually a segment of it where you have some sort of cognitive dysfunction. And mental health is a very big issue in general global health. And as you can see in this slide, 320 million patients suffer from mental disorders alone. This is a very, very low number. And you must see in this array of potential indications, diseases that we have set up here, things like substance abuse, for example, we haven't even taken up. It's definitely something that could be on this list of mental disorders. And if we put up mental disorders in this table, basically, and you look at the top row and you see the different disorders, everything from Alzheimer's to schizophrenia, you will see that we highlighted in red, and all those disorders always have a component with a deficit in cognition. And this can be very, very severe, like in Alzheimer's dementia, where your cognitive deficits are severely, severely impaired, or schizophrenia, it's the same. But you definitely also have this kind of effect in other diseases, other pathologies like depression, and even things like acute stress disorders, where you do have a deficit in cognition. And I think maybe everybody knows that for himself, that even in healthy, normal life, if you're very, very stressed, you tend to forget, misplace, or have other deficits in cognition, at least very temporarily and very briefly, but of course not as severely if you have in those disorders that are mentioned here in all the other mental disorders. The available treatments for mental disorders they are, of course, available. Not many new ones have actually been developed. And they have basically two areas where you can improve considerably. One is the time to treatment, that many of the treatments that we have currently, you actually need to treat the patient with a therapeutic agent for a prolonged time. That's the case in antidepressant, where it takes a while to develop the antidepressant effect in those patients. You have a very strong placebo effect too. And you have also a limited number of patients that actually react to a specific medication, and you actually have to try your way forward. So it's a long way until you have a therapeutic effect. And you have, of course, the risk of side effects, which in particular in compounds to treat psychosis can be very pronounced and very debilitating, actually. And you have also side effects in very compounds that have been used for a very long time, mood stabilizers, like, for example, lithium. So you have the effect, you have a protracted period to treatment, it takes a while, or you have no effect of the treatment at all. And that is more like on a try and error system, where you put on one medication. If it doesn't work, you try another one. Cognitive defects may persist even when you are being treated. And many of the drugs that are being used right now actually interact with quite a lot of neurotransmitter system in the brain. And that's where they have a tendency to also have side effects. So this is what we mean by poor pharmaceutical targeting, i.e. the specificity of the targeting is low. So the general idea is that the more specific you have your treatment, the less chances you have for having side effects. So why GABA? GABA stands for gamma-aminobutyric acid, and you have to bear with me for this acronym. It is a key player in the inhibitory system of the brain, and the brain in general, it can be seen as a balance between an excitatory or stimulating part of the brain, which is driven by a neurotransmitter system driven by glutamate, and an inhibitory transmitter system driven by the neurotransmitter GABA. And it's the balance between these two neurotransmitters, literally, that lead to a healthy brain functioning, a proper physiological brain functioning. These are the two main neurotransmitters, and of course, I've also mentioned here, we do have a lot of other neurotransmitters which are as important and very important in brain functioning. Mood disorders, for example, serotonin, the reward system driven by dopamine. But we focus on the neurotransmitter system, GABA, because we have a model of the GABA receptor in the brain, and where we can design our therapeutics, our new molecules towards this particular receptor system very specifically. So what we want to do, what we are planning to do is, with the compounds that we develop, but we want to reestablish, as you can see in this picture, there's a distorted, dysfunctional experience. What we want to do with a compound like the lead compound that we have, GT002, we want to actually reinstate the balance and functioning cognition. As you can see, that we again have an experience of the world with a functioning cognitive aspect and a reaction to the world that we actually can understand it again and function better. And this is the kind of symptoms that we try to address with the therapeutics that we develop. So what we're doing actually is we're enhancing the activity of inhibitory nerve cells. And this is not in a general way, but in a very specific subgroup. This is our hypothesis and our approach of a selective group of inhibitory nerve cells in certain parts of the brain. And by that, we want to relieve the debilitating effects of deficitary cognitive symptoms, i.e. we want to reinstate the balance, and in the previous slide, between excitatory and inhibitory neurotransmission in the brain with the drugs that we develop or the clinical candidates that we develop. So the market is responding. There's a large medical need. There's a little bit of a numbers game for how many millions or billions that this market actually is. There are CNS, of course, competing companies that develop candidates for CNS active drugs. And on the right-hand side here, I've just given you an example of what is going on in the kind of agreements that have been made targeting cognitive symptoms. This can be anything from $650, $590 million agreements between big pharma for new candidates. And these are candidates that target the GABA-A receptor system and are modulators exactly as our clinical candidates are. So this is the arena that we work in. Of course, drug development is a very difficult business. It's a very risky business because it's even with technologies and knowledge that we have now, it's not possible to actually 100% predict the effect that you will have once you get into a patient. But you can see the commercial potential that there are in this kind of projects, and we are working towards entering licensing deals, agreements in the clinical phase 1B, which we are finishing now, and doing another target engagement study in phase 2, which is then when you look at the effects in a group of patients. And the latest news was, of course, that Biogen took over a project from Sage Therapeutics, which is an American pharmaceutical company developing GABA-A receptor modulators. The most prominent one was Sage 217, zuranolone, for depression, actually. And Biogen still in-licensed this drug for an overall $1.5 billion from Sage, which had problems with this drug on a phase 3 trial, but the interest is still big enough for Biogen to take over. So this is the arena we are working in, and we think we have a new compound, actually new therapeutic options with our drugs. So we will be a first-in-class GABA-A receptor modulator. The GABA-A receptor is a very common drug target. The new thing is that our drugs target a very specific part of this receptor system, a very selective one. We have done clinical development of our main candidate, GT002. We got promising preclinical study results, I think. We have an ongoing phase 1 clinical program right now. So we're going to do some target engagement studies, and we're going to talk about it a little bit later. And then we're planning to do proof of concept, which is then studies when you go into patients and you look, for example, can you improve cognition in a certain group of patients, be it patients that suffer from schizophrenia or depression or any other indication. And I've been asked a couple of times, how do you measure improvement in cognition? And I think I have, from the right-hand side on this slide, I have a little model of some experiments we actually did. From a preclinical point of view, people have been interested in, and what you use here, and you see on the right-hand side, we do an assay called novel object recognition, and what you use is the natural curiosity of an animal, in this case a rodent, and you see on the top box, the rodent is exposed to 2 new objects. The animals are curious, and they look at the 2 objects, the one pink and one green, and 24 hours later, you give the same animals, and you change one of the objects, and since the animal is clever, it knows that one of the objects is already seen, which is the green one. And if you treat it with a normal compound, it will have it will spend the same kind of with a placebo. We will spend the same amount of time examining both objects because it's forgotten that it has already seen one of them. But if you give them a compound that actually enhances the memory and enhances the learning during those 24 hours, it will spend much more time investigating the novel object. That's why it's called a novel object recognition, i.e. what you're measuring is the memory that you have started in this animal regarding what object is new and what object has already been investigated before. So it's a little bit of a sideline on how do you measure improvement in the preclinical models. Of course, preclinical models in CNS, their translatability to the human conditions can always be discussed, but I think this model is very common, it's very standardized, and we have very nice and very good results of that, so we're very positive. We have 2 patent families on this group of compounds. We got IP protection on the key markets, and we got it actually covered until 2039. And we've got a quite extensive and very interesting approach to patent covering. So we've gone from covering not only composition of matter for our compounds and initial patents, but we've also patented the way to synthesize the compound, because we made some progress during the development of these drugs. And we actually have also applications for the way to formulate this compound, i.e. the pharmaceutical the way of administering this drugs. And we have a very good and very promising formulation, which would tend itself to a formulation that is once daily and oral, which is very, very good. And this is something that the competition, for example, I mentioned the SAGE compound, has been fighting with, and also solved it, but also after a long time. So we're very happy about that. So we've completed 2 phase 1 trials in healthy volunteers, but opened the way forward. And there's a big jump between preclinical data that we have, and once you put actually your candidate into man, to done double-blind and placebo-controlled studies. In all 48 individuals have been participating in this study. We have shown that the drug given orally is very tolerable. We haven't seen any serious side effects or any side effects at all. So the safety is very robust. The bioavailability, i.e. does the drug actually, once you give it orally, also enter the bloodstream? It's very good so-called pharmacokinetic properties. The drug is taken up, the drug is metabolized, and the drug is excreted again, which is exactly what you want to happen with a clinical candidate. So we believe that we have a very interesting pharmacodynamic profile, which now opens the way for us to go ahead. And with our clinical candidate GT002, we're going to do a so-called target engagement study. So what we're going to do is we're going to study both positron emission tomography and electroencephalogram EEG study. So what we're going to study is how does our candidate GT002 affect the electrical activity in the brain, and where in the brain it does it. This we do with a PET study, and the activity you study with electroencephalogram. And it will prove that our drug crosses the blood-brain barrier, which is of course key for CNS active drugs. We have preclinical data that shows that it does so in animals, but we of course have to prove it in humans. This study is a stepping stone, or actually a gate to the phase 2 studies, because once we got those results on our belt, then we can increase actually our interaction with the partners that we already have now, and see what kind of clinical trials or what kind of indications that they can be interested in, and go ahead with that. So it's a very interesting study, which we're going to engage next, now that we know that the drug is safe and the formulation is a very interesting one. So clinical study based on the EEG then is, once we have the profile, and we will profile it against the known ligands of the GABA-A receptor, this will be a key biomarker. So a biomarker in the sense that what does the drug do to the brain? And once you have that profile, then we can go out and see, well, that kind of profile, what would be the most interesting indication? As I said before, if we look at cognitive deficits, you can actually think of a number of indications where we have here on the left-hand side, be it schizophrenia, depression, bipolar disorder, where you can look at the effects of GT002 in normalizing the balance between excitatory and inhibitory. And that we should be able to see in the EEG. And with those data, then we can approach potential partners and say, this is our drug. It crosses the blood-brain barrier. It is safe, it's tolerable, it has a proper formulation, and we can go ahead with clinical trials, either with a partner or on our own. And of course, since we have seen no adverse effects with the GT002, it is very good that we can also test the GT002 in patients which are already on a different kind of drug regimen, because it's very difficult to find patients that are actually drug naive within the field of CNS. So what are the milestones? How did we push this project forward? You can see it in this slide. So when I think an absolute milestone was when we got the first positive results from the single ascending dose to the multiple ascending dose study in humans. And now we're marching forward towards the phase 1b study, still in healthy volunteers, in the EEG study in humans. And of course, since we have a proprietary drug model, so we're actually generating our own future drug candidates in our pipeline, which we're working on. So we are not in-licensing a drug and pushing it along the pipeline, but we're actually synthesizing our drug candidates basically from scratch, based on the model that we have developed from the basic research, which was at the very roots of Gabather from many years ago, which was academic research from the University of Lund and actually University of Copenhagen and Roskilde also. So in 2022, and we're planning initiation of a clinical phase 2 study. We're still looking at which patient group could be most interesting in that, but that will of course be based on positive results from the target engagement study and the data from the EEG and the PET study that we will get during this year. And this is a summary where we are now. We're in GT002. We've driven it all the way from discovery a couple of years ago, looking at cognitive impairment and mental disorders. We've gone all the way to phase 1b. We're now at the at the borderline, at the border area towards phase 2, where we can start thinking about patients. And it's here where we're actually looking already now, talking to potential license takers or partners, partner clinical trials, for example, and at different indications. As it's mentioned previously, that's the advantage of GT002 and the particular cognitive deficit indication, that you can look at different pathologies, different partners that might be interested in various indications, like the indications 1, 2, 3, here in a phase 2, once you look at patients. And at the same time, we still have from 2 pattern groups when we're working on preclinical development of other drug candidates. It's very important for us, both from the pattern group from GT00 compounds, that's GT00X, but we do have another second pattern group, which is structurally completely different to what we have now, and they are in the preclinical trial stage right now. And I think it's the last slide which I have here, which sort of summarizes where we are, what we have done, a little bit of our journey. We have a growing base of patients suffering from mental disorders, and actually when I made this presentation last week, I mean, I had to remember that now there will come quite a lot of new mental health issues with the long COVID and the COVID disease. The acute COVID infection might be over soon, hopefully this summer, but certainly the CNS effects of COVID and the long-term COVID effects in mental health will increase towards the end of this year and will be around for quite a while. So that's very new territory, but definitely something that we think we can contribute with therapeutic alternatives, hopefully, in things like brain fog, other CNS effects of COVID. As mentioned earlier, the available treatments are imprecise in the sense that are not selective. Some of them may cause severe side effects, and it's a question of, being able to treat or not treat, and then in some cases you're actually willing to accept some of these side effects, but there's a lot of room for improvement of the current therapeutic alternatives. So what we want to do with our selective GABA A receptor ligands is we want to optimize this treatment by being very selective, and our compounds only target the GABA receptor system. We have tested it on a lot of other systems in the brain, and it is very selective. So we do the library, and library meant in the sense of we just have a serious number of compounds of GABA A receptor modulators. They are based on proprietary research results, and with it I mean research results regarding the model that we can have to actually design the molecules that then go into our preclinical pipeline and then eventually get pushed forwards towards clinical trials. Our lead candidate, GT002, we've pushed this very hard the last couple of years to get as quickly as possible into clinical trials. We've done 2 clinical trials in man in phase 1. We're progressing this into phase 2 clinical studies. And of course, we're looking already now, talking to partners, potential people that are interested in taking license or co-development or late-stage development and commercialization of GT002. And of course, the general idea is there's a first step in a long journey to take GT002 into an agreement, and with the revenue that we can generate from that, then develop our other compounds that we have in the pipeline and get a growing company, a growing player in the field of drug discovery and drug development for CNS targeting the GABA receptor. And I think that is my very last slide, and I leave these last 6 lines to memorize for everybody. And I give it back to you.
Great. Thank you so much, Robin, for the presentation. I'd like to ask you a couple of questions. I just wanted to start by asking you if you could give us a general overview of the competitive landscape for GT002. You've already touched upon it a little, but if you could go into a little more detail about that.
Yeah, there are quite a number of players, of course, in this field. Many of them are, of course, not public information that we know of. But I mentioned the biggest one are, of course, the compounds developed by Sage. This is a group of compounds. They're within the so-called neurosteroid class. So they have a steroidal structure. They're modulators of the GABA-A receptors. And they have very different chemistry and pharmacokinetic properties to ours. But they are actually the closest that we have the compounds that are in clinical development. Now, there are a number of academic groups also that are working with molecules similar to ours, but we are covered by our patents, and they're very far away from the clinics. So we are very much ahead in this field. And people, of course, I should say, potential partners are interested in GABA-A, and with the publications and with the presentations that we've made, we've generated quite some interest. So there definitely are competitive around, not with the molecules, the type of molecules that we have, but within the target group of GABA-A receptors, definitely. And that is because that receptor system is a very good, very attractive drug target. So.
Well, great. And my next question is a little bit more speculative, let's say, but you made the connection with the mild cognitive impairment and even Alzheimer's disease as possible indications here for GT002. Of course, we know that developing a drug for Alzheimer's disease in particular has been very challenging. What are your hopes to have a drug out to treat Alzheimer's disease?
Well, we do have a collaboration with also published it with Massachusetts General Hospital. They're interested in looking at our compounds in their models of Alzheimer's disease, and definitely cognitive impairment is of course the key feature of Alzheimer's disease. And the problem with Alzheimer's disease in general is at what stage of the disease do you get in with your therapeutic alternatives? Because if you go on at very, very late stages, then it will be very difficult to achieve the therapeutic effect. But it will be very interesting if you can delay the cognitive decline in Alzheimer's disease at an early stage with our modulators. That would be absolutely fantastic. And it's again, very, very speculative. But I can tell you that the interest in modulators that improve or improve cognition, or at least sort of break and slow down the cognitive decline is enormous. This is absolutely huge, and that is because Alzheimer's disease with an aging population worldwide will be a huge issue and a huge therapeutic challenge for the future. So yeah, there are many people that are interested in this kind of modulation, because there is no real therapeutic alternative right now.
No, I'm sure. And well, just one last question before we leave. So is there one milestone in particular that you're looking forward to for 2021?
For 2021, it will be the data from the target engagement study. It will be open the gate to a lot of different options regarding clinical trials in the future. And, you know, building up a drug development company is a very long journey. I think, like in any journey, the first step is always the biggest one. And the first step for us is GT002 and the partnering and commercialization thereof. So we're very excited, because once we have that data in, could be that we've done the first step, and hopefully the first step out of many steps for the future. So it's exciting 2021 for us, absolutely.
Yeah, it sounds exciting indeed. Well, we definitely look forward to continue to following you for the next few months. And yeah, thanks so much for joining us today.
Yeah, I thank you for the opportunity to present Gabather, and we'll be in touch.
Definitely. Next up, we have the CEO of Coegin Pharma, Tore Duvold, who will be presenting a new approach to combating cancer and inflammation. Welcome, Tore.
Thank you, Michael. A pleasure to be here, and welcome to the presentation of Coegin Pharma. My name is Tore Duvold, and I'm the CEO of the company. Coegin Pharma is a Nordic biotech. It's a research-based biotech focusing on cancer and inflammation. So I will just start with a disclaimer and leave it for a few seconds, and then I will go on with the presentation. Okay, now we are ready. Coegin Pharma has a mission, and our mission is to develop a new generation of targeted, safe, effective cancer therapeutics for a number of indications where today's medicine is insufficient. Briefly about Coegin Pharma, which was introduced to the Swedish stock market last year. The company is born out of decades of fundamental and pioneering research into a superfamily of enzymes called the phospholipases. Professor Berit Johansen, our CSO, was the first to clone the most therapeutically relevant enzyme in this family, the CPLA2A alpha enzyme, which has been studied intensively in various models and in humans. This unique target has proven relevant for a range of diseases, and we have established a promising platform with opportunities in several diseases where the medical need is high. We have been able to show relevance in many types of cancer, as well as excellent effect in models for chronic kidney disease. It is also worth mentioning that the safety profile of our compounds seems to be excellent and hold the potential to become a very safe treatment. We have an experienced leadership team. I won't mention all of them, but I will highlight Professor Berit Johansen, who is our CSO and the founder. Jesper Kiel joined us last year as board member. He was formerly the corporate vice president of global regulatory affairs at Leo Pharma and personally involved in dermatology and skin cancer. We have an extensive network of international experts and CROs covering research, manufacturing, clinical operations, and corporate finance. Our operational model is very cost effective. We have virtual organization, semi-virtual organization. We have a promising drug candidates ready to go into clinical trials, and our capital need is therefore low as compared with similar companies. As we speak, we are ramping up for our phase 1/2 clinical trials in actinic keratosis, which will be concluded by the end of this year. We expect to present the results in the beginning of next year. We also plan to start a phase 2A in skin cancer next year, and we will complete the preclinical development and be ready for phase 1 next year with our systemic candidates in both cancer indications and chronic kidney diseases. We plan to spin out the two latter as separate companies in order to have the right focus and competencies. So we have an interesting therapeutic platform based on our key enzyme, the CPLA2 alpha. We strongly believe that this is a very valuable therapeutic platform with potent and selective inhibitors of the CPLA alpha enzyme. First trials come within skin diseases where we already have a clinical proof of concept in psoriasis. We have good skin properties of the drug candidate, potent inhibition of keratinocyte proliferation, a very good safety profile, and a pleasant formulation. We have seen upregulations of the CPLA2 alpha enzyme in a range of cancer cells, which is correlated with poor prognosis and mortality, and we have shown that our molecules exert potent effect on primary cell lines from cancer patients. Different types of leukemia and triple-negative breast cancer seem particularly promising. Last but not least, we have demonstrated convincing effect on kidney fibrosis in animal models. As you may know, chronic kidney diseases are often associated with diabetes, obesity, and other diseases. These are common and serious diseases, but with no effective treatment available. So what is the biological rationale and what is unique about the CPLA2 alpha enzyme? It is the key enzyme of the superfamily of phospholipases 2, regulating arachidonic acid metabolism. Arachidonic acid is central in inflammation. When something goes wrong with the immune system, triggering the phospholipase A2, it could be cytokines and stress factors, it starts overproducing arachidonic acid. Arachidonic acid is then further on metabolized to a range of pro-inflammatory mediators. So if you look at how we inhibit in this cascade, we see that we inhibit quite high in the cascade and basically blocking all the pro-inflammatory mediators. So what's happening now? This is demonstrating our pipeline, and as I mentioned before, our first trial will be in actinic keratosis. It's a combined phase 1/2 topical treatment where we will complete the study this year, and we expect the results already in the beginning of next year. We also plan our next phase 2A in basal cell carcinoma, which is a skin cancer. It's also a topical treatment where we will start in 2022 and complete the trial also next year, and with the results maybe by the end of next year or the beginning of 2023. We will soon start our preclinical program on our systemic candidate for leukemia or triple-negative breast cancer and with chronic kidney disease. When we have completed a preclinical development, we expect to be ready for initiating phase 1 in the end of 2022 and the beginning of 2023. So as you see, we have quite a lot of different diseases we're targeting with our platform. I would like to talk a little about actinic keratosis. Many people don't know it, but it's one of the most common diseases in the world. It affects a lot of people, mostly when they're middle-aged or elderly, and it's caused by overexposure of sun. Only in the US there are 60 million patients alone, and many of them are untreated. It's a precancerous cancer, and it may develop into more serious cancer. In most cases, it's treated with cryotherapy, which is a technique where you freeze the skin with liquid nitrogen, which can be painful and sometimes gives scars or cosmetic damages to the skin. There are also topical treatments. Many of them are slow acting, not so efficacious, and others can give rise to serious adverse events. So there's clearly a need for a safer and a better topical treatment that is practical for the patients to use and that works within weeks and not within months. The market value of this disease is estimated to be more than $6 billion. So a huge unmet need for new treatments in actinic keratosis. Basal cell carcinoma. This is a skin cancer. It's the most common form of cancer. And again, there is a clear unmet need. Many patients undergo surgery to remove the cancer, and sometimes it's also used cryotherapy and topical treatments. Of course, when the lesion is, the cancer is deep into the skin, you have to do surgery. But you can imagine this is also leaving scars. It's not a very pleasant therapy. And we are looking for an alternative for treating the more superficial basal cell carcinoma as an alternative to the topical treatments of today and cryotherapy. Again, the market value here is estimated to be more than $6 billion annually. So how does our trial look like? The trial itself, it's a phase 1, combined phase 1 clinical trial, and it is in itself innovative and pioneering. It's a digitally supported trial where the patients get medical support from home. We are using our AVX001, which is a potent and selective inhibitor of CPLA2, which has been proven clinically in psoriasis patients before. So this virtual trial is very patient-centric because the patients do not have to go to the hospital all the time. Of course, they do have to go to the hospital, and we're happy to have Bispebjerg Hospital, which is a leading dermatology clinic in Scandinavia, treating lots of skin cancer and other dermatological diseases every year. We have partnered up with Studies & Me, which is an innovative clinical CRO with this digital approach to the clinical trials. We are designing an app for the trial where the patients get the support from home. And this allows us to recruit patients within 2 weeks. So, focusing in on the decentralized digital clinic where you have the clinic basically at home. First, it's a single center, randomized, double-blind, vehicle-controlled dose comparison study. It's a once daily over 4 weeks, and the patients are followed 8 weeks after end of treatment. The formulation is a very pleasant gel in 2 different doses, 1% and 3%, and we compare with the vehicle, which is the gel alone, in a field-directed treatment in adult subjects. It means that you're not treating only the lesions, but you're treating a whole area of the skin. We expect to include at least 60 patients, and as always in a combined phase 1-2 trial, the primary objective is safety and tolerability. The secondary objectives are efficacy, quality of life, and dose response. We expect to run the trial from the fall and complete the trial in this year, and we expect to be able to present you the results in the beginning of next year. Just talking a little bit more about the digital trial. It means that the patients are recruited digitally, they are trained digitally, they are monitored digitally, and they can get access to healthcare specialists digitally. So they do have to go to the hospital, but most of the reporting can be done at home. It allows us to recruit patients much faster. It allows us to collect more data. It allows us to engage with the patients so that they are motivated to follow the treatment and the trial. And they feel safer because they can always get access to healthcare personnel. Our partner, Studies and Me, are specializing in this type of trials, and one of the leading ones in doing decentralized clinical trials. The next study we plan is with the same molecule, AVX001, in basal cell carcinoma. We plan to do the study in very much the same way that we are doing the actinic keratosis, but we expect that this will be a phase 2A study, a proof of concept study in basal cell carcinoma. So we're using the same documentation package, we have a similar biological rationale, and we expect to have the same fast, cost-effective, patient-centric virtual clinical trials in collaboration with Studies and Me and Bispebjerg Hospital. During this trial, we expect to have to complete the trial next year and then present the results at the latest in the beginning of 2023. So we're coming to an end, and I just want to highlight a few key points. We have a unique therapeutic target relevant for cancer and chronic kidney diseases. We have a clinical trial coming up this year in actinic keratosis, where we expect to have the results by in the beginning of next year. We are also now planning the phase 2A in skin cancer, and we expect to run that next year and have the results by the end of next year or beginning of 2023. We are seeking partners after phase 2A or eventually phase 2B with major pharmaceutical companies that can take it further and commercialize the products. Our systemic candidates for cancer and chronic kidney disease will be spun out as separate companies. So thank you very much for listening, and I think there are some questions.
Absolutely. Thank you so much, Tore, for this great presentation. Pleasure. It's great to hear that there's new approaches out there for cancer especially. In fact, that brings me to my first question, which is we normally hear a lot about immuno-oncology when it comes to cancer therapy. How does Coegin's approach differ, would you say, to immuno-oncology?
I would first say that we have seen great progress in immuno-oncology the last decade. It's not an approach that can be used for all types of cancers, and patients are also different, responding differently. So it is a very personalized treatment for certain types of cancer, which can be successful. Our approach is more on the inflammation component in cancer, basically on a very, very central enzyme, which should be a very safe way to target inflammation, it's not for all types of cancer. We have identified the type of cancer cells that are sensitive to this approach. So it's again a targeted, personalized way of treating cancer. So it's not all types of cancer. There are specific types of cancer. There are scientific speculations whether you could combine this with certain immunological principles, which is a possibility we'll look into. We also are looking into how this can be combined with more traditional chemotherapy, so it's in sort of having an add-on to standard of care. So I think that in the cancer field, we need many different approaches, and we need to be more specific on the types of cancer that we are treating.
Well, new ideas are always very welcome.
Yes, so. Certainly.
You mentioned you're collaborating with Studies and Me, this virtual CRO or digitized CRO. How does this collaboration add value to Coegin specifically?
Yeah, I followed Studies and Me since they started. They're a spin-out of Leo Innovation Lab. So I know the people. They've been working in dermatology and actinic keratosis, and they have got very, very positive comments on how they do their digital decentralized trials. So they've tried this before, but it's still pioneering. And this is the first of its kind in actinic keratosis, and we're very happy. So it's more like a partnership than a sort of a service. So we also have their scientific know-how in this, and they work very closely with Bispebjerg Hospital and Professor Merete Hedersdal, who is the leading dermatologist on this project. So a very, very good collaboration, and we're looking forward to it.
Well, just one final question for you. What are your short and long-term goals for growth in the company?
Yeah, obviously, as you see here, we're very focused on actinic keratosis and basal cell carcinoma this year and next year. We expect or hope that we will have completed these trials with a positive outcome both on safety and efficacy. So that's clearly very value adding to the company. But we're preparing for the more long term, and we have our systemic candidate, and we're looking into different types of leukemia and triple negative breast cancer with our systemic candidate, and also the very important field of chronic kidney disease. We do not think that this should be in Coegin Pharma, and we think it brings more value that we spin out these companies, and that will be good for shareholders, and it's also a way to get the right competences and the right focus. So we're very much looking forward to shape these companies and spin them out and create value long term.
Yeah, well, thanks so much for answering the questions, and again for your presentation, and we look forward to following you on.
With us now we have Tobias Thunblad, who is the CEO of MSC Nordics, as well as the chairman of the board of BioStock. Tobias also happens to be an expert on biotech valuation, and he's here today to talk about a brand new book on the subject, which he has co-written together with co-founder of MSC Nordics and current CEO of Monocle, Björn Karlsson. Tobias, very nice to have you here today. Can you start by telling me a little bit about the background of this book you've written?
Yes, so over the years we've probably done hundreds of valuations across almost any type of therapeutic area. And we've seen a lot of good examples, but we've also seen a lot of bad examples. So Björn and I, we decided that we should write a book where we actually capture best practice in how to do valuations, and at the same time create a textbook where we capture the valuation benchmarks and statistics that are useful when conducting valuations.
And many of the companies presenting here at the summit are early stage companies. When in the lifespan of a biotech company would you say it's the best time to do a valuation?
So usually I recommend that they start as early as possible. And the reason for that is that valuation, it forces you to really define your target patient population. But it also forces you to define your competitive position. And by putting dollar values on it, you will also know whether you have a return on investment or not, and if you actually should invest in a drug towards commercialization.
Yeah, and for someone who has no experience or little experience in doing these things, valuation, how would you get started?
Well, I would say that you should start simple. And the best way is to start by estimating the size of your market and the value of your business opportunity. The size of your market, the way that I would start would be to look at epidemiology. So in terms of your patient population, is it best defined by prevalence or incidence? And what target geographies do you think would be the most relevant for your addressable market? So then you have the volume of patients and the total market size. Now what you need to find out is what is the value of this. So the way that you can go about is either that you look at competing products and what they charge. If that's not possible, you could have a look at what's the alternative cost of not addressing the unmet clinical need which you are targeting.
So assessing the market size is of course important from a company perspective as well as from an investor perspective looking at a case. What other factors do you need to consider before you can sort of reach a valuation?
Yeah, so when you have your business, the value of your business opportunity, what you need to think about is the timeline. So where are you sitting right now, and what's the timeline in order for you to get to a market approval? And then you need to find out what are the studies that are needed and what are the costs of those. So when you chart this out in a spreadsheet, you will have cash flows that are initially negative and then hopefully in the end positive. And then the only thing you need to apply for this is to discount these cash flows and then sum them up. So then you will arrive at the net present value and you will know sort of the valuation right now.
And of course, it should be said that valuating a company on the fundamental data of the company and so on is quite different than valuating a stock on the stock market. But in general, what would be the key differences doing a valuation from a company from the biotech sector, for example, compared with other industries?
Yeah, so I think one of the key differences with the biotech sector is obviously the long development time that you have until you reach the market approval. Then you also have the uncertainty of not getting there. So you will, when you have a net present value, you will need to risk adjust it in some way. And there are a lot of benchmarks that are available that you can use in order to know what the likelihood of approval is from where you're sitting right now until you actually reach the market. And that can be based on your development stage, your indication category, or possibly the molecule type that you're developing.
And this all seems like a fairly complicated process altogether. How do you know that the calculated value is the right value?
Well, I would say you never know that it's the right value. And some people would say that it's more of an art than a science. However, if you make reasonable and conservative assumptions based on objective data and good statistics, then it's going to be difficult to argue that you're completely wrong. So you will have a strong foundation for evaluation, and then the rest comes down to negotiation skills.
And speaking of negotiation skills, then what would you say with your experience what are the most common mistakes that people make in these processes?
I think one of the mistakes that we see occur quite frequently is that companies start too late with the valuation process. So some companies that contact us, they already have received a term sheet from the strategic partner. And at that stage, we can do evaluations to check what the value of that term sheet will be. But the freedom to change those terms might be limited at that stage. So it's usually a better idea to do your homework first and then work proactively and probably propose the terms that you want. In our experience, you get better terms by doing it like that.
And something that often comes in focus when you see deals presented in the media, for example, that's upfront value and also the subsequent milestones, different development milestones. What can you say about these parameters in general?
Yeah, so a good way to think about it is if you have a valuation, so then you have a total value. So then the question becomes, how should you split this with the partner? How much should the biotech company get and how much should the pharma company get? And this will obviously depend a lot on what you put into your deal structure. And everything that comes early on is worth a lot more because it's not discounted as heavily. So an upfront is worth a lot more in a model like this. So if you negotiate very, very strongly that you want upfronts, then you're going to have a smaller deal size as a total deal for you as a biotech company. And it might be smart in many ways to negotiate a more backloaded deal where you have really high royalties, because that might mean that the company gets more profit in the end from the compound that you're out licensing.
That is provided that you are confident and that your asset will actually go the whole way to market, for example.
Exactly. So if you really, really strongly believe in your product, that could be a very smart way to negotiate so that you get more profit out of it in the end. And that would probably be a backloaded deal.
So in general then, as an investor looking from the outside in, so to speak, when you see a deal presented by a company that you're invested in and you can see that the upfront might not be that substantial, although the high milestones in the backload, as you say. So that's nothing to be really disappointed about then.
Well, it depends on the total structure, of course, but just having a small upfront doesn't mean that it's a bad deal. It could be a fantastic deal. The milestones could be great later on, as well as the royalties, and that's usually where the big money will come in the end.
Mm. And you said that it's all about the deal structure and difference in how you split the deal between the partners. Is there anything you can say about a general sort of scope? Where would a company be satisfied if you're going to out-license, for example, to a big pharma company? Is it, do you want, like, can you get a 50-50 deal? Can you get is it a 20-80 deal? What would be good in your opinion?
It's very case-by-case basis, but I would say companies in preclinical stage, I would say that in our experience at least, they will get somewhere between 10 to perhaps 20%, something like that, sometimes even more, whereas a clinical company might get a bigger piece of that cake. So if it's early clinical phase, perhaps somewhere between 20 to 40 perhaps, and then you can obviously negotiate even more if you're in phase 3 or later.
Mm. So later stages, higher values because it's less risk for the taker, so to speak.
Correct, yes.
Yeah, okay. Super interesting to hear about this. So my final question would be then, to conclude, how can biotech professionals and investors alike learn more about the valuation, you would say?
Yeah, so we do get this question quite a lot. Obviously, the best way is to get a lot of real-world practice. That's not always possible. So there is some literature on the subject, not a lot when it comes to biotech. That's partly why we wrote the book. But we do offer valuation courses as one way to learn more. And if anyone would be interested, feel welcome to contact me or any of my colleagues, or go to mscnordics.com and you'll find more info.
Mm. And is it also possible to buy this book as an investor, layman?
Yes, absolutely, and that will be the same thing. So just visit our website, and that will be available there.
Good concrete advice. Sorry. Thank you very much for taking the time.
Thank you so much for having me.
That effectively concludes the first day of the first ever spring edition of the BioStock Life Science Summit here from Medicon Village in Lund, Sweden. On behalf of all the team of BioStock and our event partners Nasdaq and Potter Clarkson, I'd like to thank everyone involved in today's presentations, and also of course welcome you back tomorrow at 10 a.m. For the second full day of the summit. See you soon.